Yes, students, good evening. Last class we discussed the genic level of the inheritance, and now we start with this chromosomal basis of inheritance. Chromosome basis of inheritance. It was studied by the behavior of the chromosome by Walter Sutton and Theodore Boveri. Walter Sutton and Theodore, not the behavior of the chromosome. Behavior of the chromosome was parallel to the behavior of the genes. Behavior of the chromosome was parallel to the behavior of the genes. That is a parallelism. Parallelism between chromosomes and genes was studied by Walter Sutton and Theodore Boveri. In 1902, the chromosome movement during meiosis has been worked out. It is mainly based on that the parallelism between the genes and chromosome during cell cycle or cell division. Cell cycle or cell division. And that also parallel to the behavior of genes and used chromosome movement to explain Mendel's laws. Mendel's laws. But that is comparison. Mendel's study is mainly based on genic balance, genic basis, or here it will be chromosome basis. Here it will be chromosome basis by Walter Sutton and Theodore Boveri. Okay. Then next one, that is recall you have noticed the behavior of chromosome during mitosis, equational division, and during meiosis that you have already studied in cell cycle, mitosis and meiosis, reduction division. Okay, the important things to remember are that the chromosome as well as genes occur in pairs. Chromosomes occurs in pairs and genes as well as it will also occurs as pairs. That is the first point. Genes and chromosomes occurs as pairs. That is first point. Then second point is very important. The two alleles of a gene pair. A gene consists of two alleles. A gene consists of two alleles. Okay. The two alleles of a gene pair are located on the homologous sites on homologous chromosomes. That is very important. Homologous sites on the homologous chromosome. That is very important point. That is the location of these alleles of the genes. It is located on the homologous sites on the homologous chromosome. That is another important point. Okay. That is the behavior of the chromosome during cell cycle division. That you already hear the meiosis and gamete cell formation or germ cell formation with the 4 chromosomes. With the 4 chromosomes, that is G1 phase. You have studied the G1 phase. The next S phase and G2 phase. The doubling will be takes place. Chromatids will be doubled. Okay. Then this meiosis 1, the anaphase phase. Anaphase 1 or meiosis 1. The homologous chromosomes are separate. And meiosis 2, what happens? The chromatids are separate. And each will move to germ cells or gamete cells. This is the segregation, independent segregation, or that is the case of independent pairs will segregate independently of each other. That is during meiosis, anaphase 1 and anaphase 2. Anaphase 1, the homologous chromosomes are segregate. Segregate means what? Separate. What meiosis 2 it will take place? Meiosis 2, the chromatids will separate. And it will move to each chromosome. And during this segregation or separation of this chromosome, the genes are also get segregate. The genes present in the chromosome is also segregate. That is called the parallelism. Parallelism between the chromosomes and genes, or behavior of the chromosome as well as genes. That is, it is given clear in this part here. Here the comparison between the behavior of chromosomes and genes. There is 2 columns A and B. Then what you identify here, which of these columns A or B represent the chromosome and which represent the gene. How did you decide? You go through these 3 points, and which one is the chromosome and which one is the gene. Here both will occur in pairs, either chromosome or genes. It will be either chromosome or genes. We have no idea. We finalize these things and which one is chromosome and which one is the gene. That we finally decide. Okay, you decide. How did you decide? Okay, occur in pairs. That may be either chromosome or gene. Both will occur in pairs. And segregate at the time of gamete formation, such that only one of each pair is transmitted to a gamete. Segregate at the time of gamete formation, such that only one of each pair is transmitted to a gamete. Here you see this chromosome, this pair of the chromosome, one of which will separate to a gamete. From the a pair, one of them will move to the gametes. So, chromosome segregate what is also segregate, the genes are also segregate. Genes are also segregate. But segregate at the time of gamete formation such that only one of each pair is transmitted to each pair. But single one of a pair will transmitted to a gamete. Similarly, they here also segregate at the gamete formation and only one of each pair is transmitted to gamete. Both either it will be chromosome or gene. And third point is very important. Independent pairs segregate independently of each other. Independent pairs segregate independently of each other. Independent pairs segregate independently of each other. And in the third case, here one pair segregates independently of another. This is the one pair segregates independently of another pair. That will be in the case of genes. You already go through this dihybrid cross. Dihybrid cross, dihybrid cross. You see this capital R. Capital R cross capital R, small r, capital Y, small y. Capital R, small r, capital Y, small y. Genes mark. Segregation of these characters, capital R, small r, and capital Y, small y. Capital R and small r and capital Y and small y. That will be segregate independently. One pair segregate independently of the other pair. One pair segregate independently of the other pair. That is in the case of that two genes. It will segregate independently of each other. One pair segregate independently of each other. But that will be the, here it will be gene. Here it will be chromosome. This one is the chromosome. Independent pairs of the chromosome segregate independently of each other. Independently of each other. Okay, but that is the case of others will be correct. Both are correct for genes and chromosome. Occurs in pairs segregate at the time of gamete formation such that only one of each pair is transmitted to gamete. Then independent pairs segregate independently of each other. That is in the case of chromosome. And in the case of genes, one pair segregates independently of another pair. Okay. Then next, during the anaphase stage, the diagram it is given below. During the anaphase of meiosis 1, that means anaphase 1, the two chromosome pairs are allied at a metaphase plate independently of each other. To understand, compare the chromosomes of 4 different color in the left and right columns. The left and right columns. In the left column, possibility 1. This is the possibility 1. Orange and green. Orange and green is segregate together. That chromosome segregate together, but in the right hand, here that is the case of second one, the possibility 2, the orange and red are segregating together. But that is the case of the meiosis 1, the segregation of the homologous chromosome independently of each other. Independently of each other. And anaphase 1, meiosis 2 or anaphase 2, that is the chromatids are segregated and it will move to germ cells or gamete cells. That is independent assortment of the chromosome. Independent assortment of the chromosome. That you already studied in cell cycle. It will be in cell cycle. Okay, anaphase 1 and anaphase 2. Okay. Then next it is concluded that Sutton and Bowry argued that they will argue that the pairing and separation of a pair of chromosome would lead to the segregation of a pair of factors they carry. Factors means what? Genes. Factors means genes they carry. Okay. Then Sutton united the knowledge of chromosomal segregation with the Mendelian principles. Mendelian principle means genic characters, and called it the chromosomal theory of inheritance. That's called the chromosome theory of inheritance. But Sutton and Bowry argued that pairing and separation of a pair of chromosome, separation of chromosome would lead to the segregation of a pair of factors. Factors means what? Genes. Factors means genes they carry. About chromosome segregate genes are also segregated. Sutton united the knowledge of chromosomal segregation with the Mendelian principles and called it the chromosome theory of inheritance. And you called it as chromosome theory of inheritance. Okay. Then following the synthesis of ideas, experimental verification of the chromosomal theory of inheritance by Thomas and Morgan and his colleagues. But the experimental verification of chromosome theory of inheritance was given by Thomas and Morgan and his colleagues. Led to the basis of the variation of that sexual reproduction produced. And Morgan worked with these experiments with the Drosophila melanogaster. That is called fruit fly. and which were very suitable for such studies. And what are the advantages of this Drosophila melanogaster, or it is called fruit flies? They could be grown on simple synthetic medium. Okay, they will be grown on simple synthetic medium in the laboratory, and they complete their life cycle in 2 weeks, about 2 weeks. They complete their life cycle in about 2 weeks, and a single mating or single cross, a single mating or single cross will produce large number of progenies. But that is sampling. Sampling procedure, sampling numbers or sample numbers are very huge number. Single mating would produce a large number of progeny flies. Okay. Then also there was a clear differentiation of the sexes, sex demarcation or differentiation of the sexes. The male and female flies are easily. The male flies are smaller and female flies are larger. The sex demarcation. That will be male and female flies are easily distinguishable or demarcated. It will be demarcated. And also there are many types of hereditary variations noted in the low power microscope. So there will be several hereditary variations also reported. That is the important characteristics of Drosophila melanogaster, which will be most suitable for the studies of T.H. Morgan to the experimental verification of chromosomal theory of inheritance. Our chromosomal theory of inheritance was put forward by Walter Sutton and Theodore Boveri, and that is experimental proof for that theory will be chromosome theory of inheritance was put by discovering the basis of variation T.H. Morgan. Okay. Then linkage and recombination. Next studying is very important. Linkage and recombination. That is studied Morgan in Drosophila melanogaster, and that will be very important experiment he conducted. He got Nobel Prize for that study. T.H. Morgan. Morgan carried out several dihybrid crosses in Drosophila to study the genes that were sex-linked. That will be sex-linked. That characters he studied, 2 characters studied, that will be sex-linked, or the genes are get linked. Sex-linked. And the crosses were similar to dihybrid crosses carried out in Mendel in peas. About dihybrid cross conducted by Mendel in garden peas. Similar to that, in Drosophila, T.H. Morgan will do that dihybrid cross. Okay. For example, an example for this study of dihybrid cross in Drosophila melanogaster, Morgan hybridized yellow body and white-eyed females. So that is the 2 characters he took will be the body color and the eye color. Yellow body and white-eyed females. Here, clear sex demarcation is here. Okay. Well, that is in the case of yellow body and white-eyed females are crossed to brown body and red-eyed males. Brown body and red-eyed males. That we called brown body and red-eyed males. They are called wild types. Brown body and red-eyed. Well, here, yellow body and white-eyed females to brown body and red-eyed males. They are the parentals. And intercross their progeny, F1 progeny. But result between the yellow body and white-eyed females to brown body and red-eyed males. And they get F1 progeny. Then F1 progenies are intercrossed. What is the result in the F2? What is the result he observed? Morgan observed that 2 genes did not segregate independently. That 2 genes did not segregate independently of each other. And F2 ratio deviated very significantly from 9:3:3:1 ratio. That is, F2 ratio will be very significantly varies. Deviated means varies from 9:3:3:1. There will be no independent. There will be 2 genes did not segregate independently. And what happens here? Here the 2 genes are get linked. Physical association of the genes. Expected when the 2 genes are independently assorted, but it is not independently assorted because the ratio is deviated. F2 ratio is deviated from 9:3:3:1. Then he undergoes some conclusions. Morgan and his group knew that the genes were located on the X chromosome. Section 5.4 clear right. And so quickly What when the two genes in a dihybrid cross were situated on the same chromosome? Quickly, that when the two genes in a dihybrid cross were situated on the same chromosome, the proportion of the parental gene combination was much higher than the non-parental type. But he took the parental types of yellow body and white eyed, and another parent is brown body and red eyed. And that number, the proportion of the parental gene combination were much higher than the non-parental types. What are non-parental types? Yellow body and red eyed. Yellow body and red eyed. And next one, brown body and white eyed. Brown body and white eyed. That is called non-parental gene combination. Non-parental gene combination. That is very important. What here took place, mostly that in this cross, it will be the linkage. The genes are physical association of the genes, and it is called linkage. Morgan attributed this is due to the physical association or linkage of the two genes and coined the term linkage. Coined the term linkage to describe what? This physical association of the genes on a chromosome. On a chromosome, that is called a linkage. And what will be the result? They will lead to proportion of the parental types will increases. Parental types will be increases. Parental types will be increases. That is very important. Okay. Then what is meant by the term recombination? The term recombination to describe the generation of non-parental gene combination. This recombination that will leads to formation of non-parental combination. What it is called recombinance. It is called recombinance. Parental, non-parental forms are called recombinance. But here it is the case of his studies in Drosophila, that is, he hybridized the yellow body and white-eyed females and brown body and red-eyed males, and the F1 progenies are intercrossed, and in the F2 ratio, the data will be very significantly deviated from 9:3:3:1 ratio. Where he expected when the two genes are independent. Independent work started, but it is not. Genes are not independently segregated. And here the main reason is that is due to a much higher than the proportion of the parental gene combination were much higher than the non-parental type. Over here, the Morgan attribute is due to the physical association, or it is called linkage of the two genes. Linkage of the two genes, and coined the term linkage to describe the physical association of genes on a chromosome. And the term recombination to describe generation of non-parental gene combination. Non-parental. Recombinance. It's called a recombinance. Okay. So that is parental and non-parental. Parental types are yellow body and white-eyed. Yellow body and white-eyed. And other one is brown body and red-eyed. Brown body and red-eyed. Non-parental forms are what? Yellow body and red-eyed. Yellow body and red-eyed, and brown body and white-eyed. Brown body and white-eyed. These are the parental ones. These are the parental ones, and these are the non-parental ones. Okay. Then Morgan and his group also found that when the genes were grouped on the same chromosome, some genes were very tightly linked. If the two genes located on the chromosome very close to each other, two genes are located on the chromosome very close to each other, that will be tightly linked, or it will be close association, physical association of the genes, tightly, and there will be no chance of crossing over or recombination. There will be no recombination. Some genes were tightly linked and showed very low recombination. About any chance there will be no chance of recombination. Very low recombination. That is called tightly linked, or they will show linkage, clearly linkage. Very low recombination. Okay. Then cross cross while others were loosely linked. Loosely linked means what? Showed higher recombination. If the two genes located very farther from each other, then there will be a chance of crossing over. There will be a chance of crossing over, or that is the case of recombination. There will be a chance of recombination. About that, what happens? Here it will be loosely linked, showed higher recombination. They will show the higher recombination. There will be chance of recombination, higher recombination, and linkage strength will be reduced. That is figure 5.11 cross B. But two crosses we next page figure 5.11 cross A and cross B. Loosely packed on linkage linkage recombination strength closely packed closely packed very closest to each other. There will be a physical association of genes and there will be no recombination. And it will show the low recombination, very low recombination. The result studies are Morgan study, linkage and recombination. These two concepts are very important, linkage and recombination. Okay. The next point is very important. For example, found that the genes white and yellow, first clause white, that is a white eye color and yellow body were tightly linked and showed only 1.3% recombination, while white and miniature, second cross, showed that 37.2% recombination. So 1.3% is very minute recombination. 37 very large recombination. Very large. His student Alfred Sturtevant used the frequency of recombination between gene pairs on the same chromosome as a measure of the distance between the genes and mapped their position on the chromosome. About his student, that is Morgan student. Morgan student Alfred Sturtevant used the frequency of recombination between gene pairs on the same chromosome as a measure of the distance between the genes, and they are mapped their position on the chromosome. That's called chromosome mapping or gene mapping. It is called chromosome mapping. And how he found out this frequency of recombination or recombination frequency? He conducted the test and that is called test cross, and he find out the recombination frequency. How we find out the recombination frequency? Recombination frequency means number of recombinants or non-parental forms. Number of recombinants divided by total number of progenies. Total number of progenies into 100. That is recombination frequency. And how we calculate use the frequency of recombination between gene pairs and on the same chromosome as a measure of the distance between the genes. How we convert this recombination frequency? There is the simple conversion. 1% recombination frequency is equal to 1 map unit. 1% recombination frequency is equal to 1 map unit. Then if it is the recombination frequency 25%, what is the map unit? There will be 25 map units. Or it is called Morgan unit, or it is called Senji-Morgan. That unit is also Senji-Morgan or Morgan unit. And mapped their position on the chromosome. Okay, but that is the gene mapping or chromosome mapping by Alfred Sturtevant. He is a student of Morgan. Okay, then next very important point, that is the cross, it will be done. And that gene mapping are extensively used as a starting point of sequencing of old genomes in the case of human genome sequencing project. Human genome project sequencing, gene sequencing. Gene mapping or the chromosome mapping we used. Okay. Then the cross A and cross B, that is Morgan's cross, that is connected with the linkage and recombination. Cross A, he will first cross the parental ones, yellow body and white-eyed females. These are females and these are males. That's to X chromosome. Here it will be X and Y chromosome. This one is X and this one is the Y chromosome. And these genes are located for body color and eye color is located on the X chromosome. X chromosome. Drosophila, the sex determination mechanism is XX, XY mechanism. It will be XX and here it will be XY. XY. These are the females and these are the males. Males and females. Okay. Then yellow body and white type. Yellow body and white type females are crossed with the wild type males. Wild type males means brown body and red type. Okay. This is the cross. And the F1, what do you obtain? All the females are wild type. And males are yellow body and white eye color. That clearly shows that these two characters are, that is body color and eye color, are located on the sex chromosome. It will be in the sex chromosome, and they will also say that the chromosome, both the genes are located on the X chromosome. X, sex and X chromosome. Okay. Then intercross is the F1 progenies. F1 progenies, intercrossed, F1 generation, the progenies, that is wild type females and yellow body and white type males. Then what happens? What is the result in the F2? That will be significantly deviated from 9:3:3:1 ratio, because here the parental types are showing 98.7%. Parental types are 98.7% and recombinants are 1.3%. That is, recombinant means non-parental types. Non-parental types. So that is in the F2 generation. Intercrossing these F1s. Intercrossing the F1s. F2 will show this parental percentage. That is, both males and females in the all these one are parental types, and it is 98.7%. 98.7%. And the non-parental types, or that is called recombinant types, will be only 1.3%. So here the wild type males and the yellow body and white head females, then wild type females and the yellow body and white head, females. Both are males and both are females. All are showing the parental types, and it will be 98.7%. And here it is in the case of the brown body and white head, brown body and white head, and yellow body and red head. Here also brown body and white head and yellow body and white head. Brown body and white head. Here the yellow body and white head. Yellow body and white head. These will be, all these will be the recombinants. They are called recombinants or non-parental. It will be only 1.3%. So that means what? That is the case of the recombination percentage is very less, and linkage will be more. Linkage strength will be more, and recombination will be less. That is in the case of first cross. That is the first cross. That is cross A. Linkage, result of two diameter cross conducted by Morgan. Cross A shows the crossing between gene Y and W. Y for body color and W for eye color. Cross B shows crossing between genes with the W and M. That is eye color and wing size. Eye color and wing size. Wing size. Here, dominant wild type alleles are represented with the superscript plus. Superscript plus. Then note the strength of linkage between Y and W. Y and W, the strength will be 98.7%. That will be strength of linkage will be higher. In the case of first cross, that is Y and W. Whereas than the W and M. W and M, here the recombinant percentage will be 37.2. But here 1.3%, here 37.2%. That means here more recombination takes place and linkage strength will be decreases. That is, the strength of linkage between Y and W, this cross will be higher than W and M. So that is in the case of the cross between this dihybrid cross in Drosophila by T.H. Morgan. So cross F1 generation, F2 generation, okay, and parental ones, yellow body, white head, and wild types. And here the F1 generation, the females are wild type and males are yellow body and white head. And the intercrossing of this F1 generation, then the F2 will produce. All the most of them are 98.7% are parentals, and only 1.3% are the recombinants. That clearly shows that it will be what? It will be linkage will take place. Physical association of the genes will take place, and independent assortment or recombination will be very less. Whereas in the case of the second cross, that is body, that is eye color and wing size, miniature wing and normal wing, wings. Here in the case of the recombinant types, will be increases from 1.3 to 37.2, and the parental type will be decreases from 98.7 to 62.8%. That means the linkage strength is less in cross B compared to cross A. So here the cross A, the linkage will mostly takes place, almost 98.7%. That means completely linked. Here it will shows only partial linkage. It will shows only partial linkage, that is second cross. Okay, so that is about the linkage. That you just draw that diagram of that cross, and you go through that diagrams especially. Okay, then next polygenic inheritance. Next inheritance is called polygenic. What is meant by polygenic? More than two genes will, or three or more. More than two means three. Three or more genes will control a character. It is called polygenic inheritance. Polygenic inheritance. More than three. Three or more than three. Three or more than genes will control a character. That is called polygenic inheritance. Here it is an example. Mendel's studies mainly describe those traits that have distinct alternate terms, forms such as flower color, which are either purple or white. Here Mendel's studies, it will be alternate, that is distinct alternate forms like white, purple or white, or violet or white. But if you look around, you will find that there are many traits which are not so distinct in their occurrence and spread across a gradient. Spread across a gradient. Here it will be discontinuous, purple or white, round or wrinkled, or yellow or green. That will be discontinuous or their distinct alternate forms. But most of the characters, traits you studied in the nature, they will be, the occurrence will be spread across the gradient. That means continuous variation. The variation will be continuous. For example, in humans, we don't just have tall or short people. Either tall or short. No. There will be as two distinct alternatives, but a wide range of possible heights. Here, we don't just have tall or short people as two distinct alternatives, but a wide range of possible heights. Wide range of possible heights. Okay. Such traits are generally controlled by, that type of traits are generally controlled by three or more genes. Three or more genes, and that's called polygenic traits. And it is called polygenic. It is controlled by three or more genes and are called polygenic traits or polygenic inheritance. That inheritance are called polygenic inheritance. Besides the involvement of these multiple genes, polygenic inheritance also takes into account of the influence of environment. That polygenic inheritance or more than three genes will control a character, that will also take into account of the influence of environment also. There will be a polygenic inheritance controlling the process or the interactions. Here the influence of environment is also having a factor. Environment factor is also influencing. Okay. The typical example for this polygenic inheritance is human skin color. Human skin color. Human height is one example. Human skin color is another example. Classic example for this. In a polygenic trait, the phenotype reflects the contribution of each allele. In a polygenic trait, the phenotype reflects the contribution of each allele. Contribution. Each allele is contributed. That phenotype. That means the effect of each allele is additive. Additive means what? Cumulative. It will be cumulated. If it is more dominant alleles are cumulated, that will move to a phenotype. If more recessive alleles will accumulated, or that will be in the case of additive, or it is called contribution. Contribution of these alleles, cumulative contribution. It is called additive inheritance. This effect of each allele is called additive, and that inheritance is called additive inheritance. Okay. Then a better example to understand this. That is the contribution of each allele that will affect the phenotype. That will be the three genes A, B, C control human skin color. So human skin color is controlled by three genes. What are the three genes control human skin color? A, B, and C. Skin color, that is the melanin production. Melanin production in human skin. Human skin color melanin production. It is under the control of three genes, that is capital A, capital B, capital C. Control the skin color in human with dominant forms capital A, capital B, capital C, responsible for the dark skin color. Dark skin color, that is extremely dark or black, extremely dark color. Extremely dark color or black color. Okay, then next. And the recessive forms of these genes A, B, C, small a, B, C, small a, B, C, small a, B, C, for light skin color. Light skin color. But that is the contribution of this allele. If it is all the dominant alleles will come, it will be gives more dark. If all the alleles will be recessive ones, it will be light. That will be addition of these alleles or contribution of these alleles. Contribution of these alleles will give the phenotype will be changed. Phenotype will be changed. The genotype with all the dominant alleles. Dominant alleles, all the dominant alleles what? Capital A, capital A, capital B, capital B, capital C, capital C. That will give the darkest skin color. Capital A, capital A, capital B, capital B will have the darkest skin color. darkest. It is extremely black. Extremely black. Skin color. And with all the recessive alleles. All the recessive alleles means what? Small a, small a, small b, small b, small b, small e. That is the lightest skin color. Lightest, that is extremely white. But that is the, in population, darkest ones and lightest one, or extremely black ones and extremely lighter ones. Small a, small a, small b, small b, small c, small c. Will have the lightest skin color. Then what expected in the genotype with the 3 dominant alleles and 3 recessive alleles? 3 dominant and 3 recessive alleles. That is capital A, small a, capital B, small b, capital C, small c. What is the skin color? Here the skin color is intermediate skin color. Intermediate, not extremely dark or extremely white. There will be intermediate skin color. Intermediate skin color. Okay, so there will be a continuous variation here. That 3 dominant and 3 recessive alleles will have an intermediate skin color. In this manner, the number of each type of alleles in the genotype would determine the darkness or lightness of the skin in an individual. So either it will be capital A, capital A, capital B, capital B, capital C, that will be darkest or extremely dark persons, and small a, small a, small b, small b, small b, the lightest persons, lightest person. Okay, and that is done by an important scientist, that is in human populations, to important human population by C.B. Davenport. Davenport will study this experiment. That is in the case of he will took the 2 population, that is Negroes from Jamaica, and Caucasian, extremely white persons from Buddha, Malaysia, Buddha, and there will be a cross, or there will be marriage between these Negroes and these ones are called Caucasians. And F1, that is first generation, they are intermediate skin color, and they are called mulattoes. The intermediate or hybrid ones are called mulattoes, and a marriage between the mulattoes in the F2 ratio. That will be showing very variation. That variation will show 1:6:15 in the F2 ratio. 15:20:15:15:6:1. That is the ratio. There will be a variation. Clear cut variation. 1:6:15. 1:6:15:20:15:6:1. This is extremely black, this is extremely white, these are the intermediate. Then it will be fairly black, or that will be intermediate, or dark, white, fairly white or white, then it will be intermediate. But there will be continuous variation. There will be continuous variation in the case of polygenic inheritance. Human skin color. Over here there is a contribution of these alleles that will give to additive influence to the skin color. Okay, so this is the polygenic inheritance. More than 3 or more than 3 genes. 3 or more than 3 genes will control a trait. That is called 3 or more genes are thus called as polygenic trait. Okay. Then next one, another concept you have to study will be the pleiotropism. Next one is pleiotropy. We have so far the effect of a gene on a single phenotype or trait. Now, we so far seen the effect of a gene on a single phenotype. There will be a single gene will control a single trait or phenotype. However, the instance where a single gene can exhibit multiple phenotypic expression. A gene will control more than one phenotype. That is called a multiple phenotypic expression. Or manifold effects of a gene. Such a gene is called a pleiotropic gene. Okay. The underlying mechanism of pleiotropy in most cases is the effect of a gene on metabolic pathways which contribute towards the different phenotypes. Example, that is the case of phenylketonuria. That is called phenylketonuria, PKU, which occurs in humans. The disease is caused due to the mutation of gene that codes for the enzyme phenylalanine hydroxylase. So that is pleiotropy. Here it is a typical example is phenylketonuria. That is shown in humans is called phenylketonuria. It is a disorder, metabolic disorder. Or it is called inborn error. It is called the inborn error of metabolism. That we study later. Metabolism. Okay. And here it will be in the case of the disease is caused by the mutation in the gene that codes for the enzyme phenylalanine hydroxylase. That is single gene mutation. But here in the case of this metabolic pathway, the amino acid phenylalanine is converted to tyrosine. Phenylalanine is converted into tyrosine with the help of an enzyme phenylalanine hydroxylase enzyme. Phenylalanine hydroxylase enzyme. And this enzyme is lacking due to the mutation in the gene that codes for the enzyme phenylalanine hydroxylase. That is single gene mutation. And this gene is not working, the enzyme is not produced, and there will be no conversion to phenylalanine to tyrosine. And this phenylalanine is accumulated, and it will convert into phenylpyruvic acid and other derivatives. Phenylpyruvic acid and other derivatives, and it will be accumulated in brain and forms mental retardation. And this pathway from the phenylalanine to tyrosine, and tyrosine finally enters another pathway to melanin, the production of melanin, tyrosine to melanin production. That melanin will give the skin coloration to hair and skin. Hair and skin coloration, pigmentation melanin pigment. About here, the melanin production, that is also from the same pathway. That is phenylalanine is converted to tyrosine by phenylalanine hydroxyl, then tyrosine to melanin. Up here, this conversion is not takes place, and this conversion, that is tyrosine to melanin conversion, is also not takes place. There will be, that is the case of this pathway will be, error will be seen in that pathway. That will also leads to the reduction in hair and skin pigmentation. But this manifests itself through the phenotypic expression characterized by mental retardation and a reduction in hair and skin pigmentation. Where a single gene will control two phenotypes or two characters or two traits. That is called pleiotropism. That example is called pleiotropism. A typical example for pleiotropism is phenylketonuria. Here, phenylketonuria, this will leads to the either the mental retardation and reduction in the hair and skin pigmentation. That is another one, pleiotropism. The next one is sex determination. Next heading. Okay. The mechanism of sex determination is also a very puzzle for the genetist. And it will be studied by some cytological observation by some scientists in insects. And they will study the concept of genetic or chromosomal basis of determination in cytological observation. Chromosomal or that is genetic basis of sex determination in some insects. And it will be the cytological observation. And NKEAM, a scientist called NKEAM in 1891, would trace a specific nuclear structure through spermatogenesis, the formation of male gametes, sperms. Spermatogenesis in a few insects, and it was also observed by him that 50% of the sperm receive this structure, that is called the specific nuclear structure obtained in the 50% of the male gametes, and whereas other 50% sperms did not receive this special nuclear substance. Special nucleus or specific nuclear structure is not obtained in the 50% of the male gametes, and 50% they will get a specific nuclear structure is obtained. And NKEAM gave the name to this structure is called X body. He called that 50% of the male gametes are having some special nuclear structures, and they are called him as X body, and other 50% having no that X body. But he could not explain what is the X body and its significance. Then further investigation by other scientists led the conclusion that X body of NKEAM was in fact a chromosome. Later, NKEAM chromosomes identify and further investigation by other scientists concluded that the X body of NKEAM was in fact a chromosome. X body that will be a chromosome, and that is why it was given the name X chromosome. That later the scientists will coin that X body of NKEAM is given X chromosome. Name will be X chromosome. X body will be changed to X chromosome. It was a chromosome that is later studied. Okay, it was also observed that large number of insects, the mechanism of this sex determination is of the X0 type. In the case of sex determination in most of the or large number of insects will be X0 type of sex determination. That sex determination is called a X0 type of sex determination. X0. Here all in the case of X0 type of sex determination, the females have all the females, female X bear an additional X chromosome. There will be two X chromosome besides the other chromosome. Besides the other chromosome means what? Autosomes. Besides the autosomes, two X chromosomes in females. On the other hand, the males, what happened? 50% of the males are having that's X chromosome. That is the special body that's called X chromosome. And other 50% having X. 50% of the male gametes are having X chromosome, and other 50% having no X chromosome. No X chromosome. Without X chromosome, or it will be zero. It will be zero. That is, it is given here. On the other hand, some of the sperms bear the X chromosome, whereas some do not. Then X is fertilized by sperms having an X chromosome. So here it will be when X with another male gamete with the X. That will be become females. This will be a female. And those fertilized by sperms without X chromosome. Without X chromosome, with the female X chromosome, it will be X0, and they will be male. Okay, but that is in the case of X fertilized by sperms having an X chromosome become females, and those fertilized by sperms that do not have an X chromosome become males. That is called X0. Do you think the number of the chromosome in the male and female are equal? No. The male is less than one chromosome than the female, because there will be X0 besides the autosome. Autosome will be same. Autosome number will be same, but there will be lack of one X chromosome. In males. In the case of males compared to females. Okay. Then next, due to the involvement of X chromosome in the determination of sex, it was designated to be the sex chromosome. X chromosome it's called a sex chromosome. It is designated as the sex chromosome. And the rest of the chromosome were named as autosome. Body chromosome, that is called a autosome. Okay. Autosome plus XX in the case of female. Autosome plus X0 in the case of males. That is the X0 sex mechanism. That is especially in insects, large number of insects. Especially typical example for this X0 mechanism is grasshopper. Is an important example of X0 sex determination. Males have only one X chromosome besides the autosomes, whereas females have a pair of X chromosome. Okay, that is about the first sex determination, that is called the X0 mechanism. Then second, അടുത്ത mechanism നമ്മൾ പഠിച്ചിട്ടുള്ളതാണ് നമുക്കറിയാം, that is XY mechanism. About that observation will lead to a number of species to understand the mechanism of sex determination. In a number of other insects and mammals, including man, what is the type of sex determination? That is called a XY type of sex determination. XY type of second type of sex determination, that is called a XY type of sex determination. Other insects and also mammals, including humans. XY mechanism, that is called XY type of sex determination, in which both male and female have same number of chromosome. Among the males, an X chromosome is present, but its counterpart is distinctly smaller and called the Y chromosome. About X chromosome idanengil Y chromosome is smaller than the X chromosome. X chromosome. That is in the case of Y chromosome. About X chromosome is present, but its counterpart is distinctly smaller and called the Y chromosome. That is called XY in males. Whereas in females, however, have a pair of X chromosome. Okay, but that is in the case of XY sex determination mechanism. Here, both males and females bear same number of autosomes. Plus autosomes. Same number of humans like autosomes 44A. 22 pairs of autosomes. 22 pairs. 44 autosome plus XX or 44 autosome plus XY. That is in the case of male. In the case of humans. Okay. Here, hence the males have autosome plus XY, while females have autosome plus XX. Autosome plus XX. But there are 23 pairs in human beings. 22 pairs are autosomes and 1 pair is sex chromosome. Okay. But there is XY sex determination mechanism. XY sex determination. Hence, the males have autosome plus XY while females have autosome plus XX in human beings and in Drosophila. Drosophila also having the same sex determination. The XY male and a XX female. XY male and a XX. And humans also XY male and a XX female. XY male and a XX female. Drosophila and humans also having the same system or same type of sex determination. Okay. Then in human beings and in Drosophila, the males have one X and one Y chromosome, whereas females have a pair of X chromosome besides the autosome. Okay. Then next, in the above description, you have studied about two types of sex determining mechanism. That is, one is called X0 type, another one is called XY type. One is X0, first one. In most of the insects, or a typical example is grasshopper, and the XY type, that is other insects like Drosophila and all the mammals including humans, there will be XY type. But in both cases, X0 type and XY type, in both cases, X0 and XY, both cases, males produce two different types of gametes. Males produce two. In the case of X0 type, what is the two types of male gametes? Either with or without X chromosome. In the case of X0, it will produce either X or without X. Either X or without X. And in the case of type B is what? XY. B. Some gametes with the X chromosome and some with the Y chromosome. Some with the X chromosome and some with the Y chromosome. X0 and XY. X0 X chromosome without X chromosome. 0. Here X and Y. This is XY. X and Y. Here one with the X and one with the Y. Two types of gametes are produced. Two types of gametes. That is, some gametes with the X chromosome and some with the Y chromosome. These are gametes. These are the gametes. X0. That will be X0. These are the gametes. Okay. Such a type of sex determination mechanism is designated to be the example of male heterogamety. Male heterogamety. That is, male produce two types of gametes and it is called male heterogamety. Male heterogamety. In some other organs, example birds. Birds, another mechanism in birds. Here, the different mechanism of sex determination is observed. In this case, the total number of chromosomes is same in both males and females. Total number of chromosomes will be same in both males and females, but two different types of gametes in the terms of the sex chromosome in the case of females. In this case, the total number of chromosomes is same in both males and females, but two different types of gametes in the terms of the sex chromosome are produced by females. So females are produced two types of gametes. Two different types of gametes. In the case of female, two different types of gametes are produced in the case of females. That is called a female heterogamety. It is called a female. In the case of X0 and XY mechanism, males are producing two types of gametes, whereas females are having homogamety. In the case of birds, the females are produced two types of gametes and it is called female heterogamety. In order to have a distinction with the mechanism of sex determination described earlier. The two different sex chromosomes of a female bird has been designated to be Z and W. For female, Z and W, ZW. Not X and Y, it is given as ZW. That is to remove that confusion. Z and W chromosome. Okay, in this organism, the females have one Z and one W chromosome. One Z and one W chromosome. Whereas males have a pair of Z chromosome. Males have a pair of Z chromosome. And females have two types of sex chromosome, that is Z and W, besides the autosomes. Autosomes. That is in the case of the third sex determination, that is called ZZ-ZW mechanism. It is called ZZ-ZW mechanism, that is birds. Here the males are homogametic and females are heterogametic. Females are heterogametic. That is Z and W. But females will control the sex. Females will govern, or females will determine the sex. In the case of birds, males are homogametic. Males are having same chromosome set. Females are different, set W. Here the males are XY. In the case of Drosophila, humans, etc. Males are heterogametic and females are homogametic. In this case, the birds, it is called female heterogamety. It is called female heterogamety. Female heterogamety in birds. And females will control sex. In the case of Drosophila, male heterogamety. Male heterogamety. In human also there will be male heterogamety. Humans also there will be male heterogamety. Okay, so these are the three types of sex determination mechanism. Three types of sex determination mechanism. Okay, the next one, sex determination in humans. നമ്മൾ ഓൾറെഡി പഠിച്ചു കഴിഞ്ഞിട്ടുള്ളതാണ് ഹ്യൂമൻസിൽ സെക്സ് ഡിറ്റർമിനേഷൻ നമ്മൾ പറഞ്ഞു XY type ആണ്. Out of the 23 pairs, 22 pairs are exactly same in both males and females, and these are called autosomes. And a pair of X chromosome is present in female, and males there will be one X and one Y chromosome. Okay, during spermatogenesis among the males, two types of gametes are produced. 50% of the sperm were X chromosome, and 50% of the sperm have Y chromosome besides the autosome. Then females have produced only one type of ovum with the X chromosome. Then there is an equal probability of fertilization of the female ovum egg with the male X chromosome that will give rise to there will be a female XX, and fertilization of ovum with the Y chromosome will result into males. About for every pregnancy there will be a chance of male and female, that is 50-50 ratio. 50% male or 50% female, that will result into thus it is evident that in genetic makeup of the sperm determines the sex of the child. എന്താണ് മെയിൽസിന്റെ സ്പേമിനെ നോക്കിയിട്ടാണ് സെക്സ് ഡിസൈഡ് ചെയ്യുന്നത്. അപ്പോൾ മെയിൽസിന്റെ രണ്ട് തരം ഗാമറ്റ് ആണ് എക്സ് ആൻഡ് വൈ. അപ്പൊ എക്സ് വിത്ത് ഫീമെയിൽ എക്സ് ആണെങ്കിൽ അത് ഫീമെയിൽ ആയി, എക്സ് വിത്ത് വൈ ആണെങ്കിൽ എന്തായി, വൈ വിത്ത് എക്സ് ആണെങ്കിൽ ദാറ്റ് വിൽ ബി മെയിൽ ആയി, അല്ലെ മെയിൽ. അപ്പൊ സെക്സ് ഡിറ്റർമൈൻ ചെയ്യുന്നത് മെയിൽ ആണ്, അത് നമുക്ക് മനസ്സിലായി കഴിഞ്ഞു അല്ലെ. എവിഡന്റ് ദാറ്റ് ദ sperm that determines the sex of the child, നമുക്ക് അറിയുന്ന കാര്യമാണ് അതൊക്കെ. It is also evident that in each pregnancy there is always 50% chance probability of either male or female child. അതുകൊണ്ടാണ് നമ്മൾ അതിൽ ചില കേസുകൾ അവിടെ എഴുതി വെച്ചിട്ടുണ്ട്. അൺഫോർച്ചുനേറ്റ് ദാറ്റ് ഈസ് അവർ സൊസൈറ്റി, വുമൺ ആർ ബ്ലെയിംഡ് ടു ഗീവ് ബർത്ത് ടു ഫീമെയിൽ ചിൽഡ്രൻ, അല്ലെ. പെൺകുട്ടികളെ പ്രസവിക്കുന്ന അമ്മമാരെ, അല്ലെ, അല്ലെങ്കിൽ അവരെ നമ്മൾ മാറ്റി നിർത്താണ്, ജനങ്ങൾ മാറ്റി നിർത്താണ്, അല്ലെങ്കിൽ അവരെ ഒരു പ്രത്യേക ഒരു ഇതിൽ അവരെ till it will be ostracized and ill treated, അവരെ ill treated ആണ്, അല്ലെ. അവരെ നമ്മൾ ട്രീറ്റ് ചെയ്യുന്നില്ല, അവരെ വളരെ മോശമായിട്ടാണ് ട്രീറ്റ് ചെയ്യുന്നത്. അങ്ങനെ കുറെ കേസുകളൊക്കെ ഉണ്ട്. നമ്മൾ ഫീമെയിൽ ഇൻഫാന്റിസൈഡ് ഒക്കെ നടത്തിയിരുന്ന സ്ഥലങ്ങൾ, ഉറുസ്ലാംപെട്ടി തമിഴ്നാട്ടിലൊക്കെ ഫീമെയിൽ ഇൻഫാന്റിസൈഡ് വളരെയധികം കൂടിയിട്ടുള്ളതിന്റെ കാരണങ്ങളൊക്കെ ഇതാണ് മെയിൻ കാരണം, അല്ലെ. Okay, അപ്പോൾ that is sex determination in humans, that will be very common. Then next another feature it will be in the case of sex determination, special type of sex determination, honeybees. ഹണിബീസില്, വാസ്പില്, അതുപോലെ തന്നെ ആൻസില്, അല്ലെ, ഉറുമ്പുകളില്, ആൻസില്, അതുപോലെ തന്നെ ഹണി ബീസിലേക്ക് സ്പെഷ്യൽ ടൈപ്പ് ഓഫ് സെക്സ് ഡിറ്റർമിനേഷൻ ആണ്. അതിനെ നമ്മൾ എന്താ വിളിക്കുക? ദാറ്റ് ടൈപ്പ് ഓഫ് സെക്സ് ഡിറ്റർമിനേഷൻ ഇസ് കോൾഡ് ഹാപ്ലോഡിപ്ലോയിഡി. അതിനെ നമ്മൾ വിളിക്കുന്ന പേരാണ് ഹാപ്ലോഡിപ്ലോയിഡി. Haplodiploidy. Here the sex determination honeybee is based on the number of sets of chromosome. Number of sets of chromosome in an individual receives. അത് ബേസ് ചെയ്തിട്ടാണ് ഇപ്പൊ നമ്മൾ പാരന്റ്സ് എടുക്കുകയാണ്, ഫീമെയിൽ ആൻഡ് മെയിൽ. Sets of chromosome നോക്കിയേ. The offspring formed from the union of a sperm and an egg develops as a female queen or worker. And അപ്പൊ female or queen or worker that is having diploid to an 32 chromosome. The set of chromosome will be 32. Okay. And an unfertilized egg. ഇതാണ് ഫീമെയിൽ പ്രൊഡ്യൂസ് ചെയ്യുന്ന എഗ്, അല്ലെ. ഫീമെയിൽ പ്രൊഡ്യൂസ് ചെയ്യുന്ന എഗ്, അല്ലെ. ആ എഗ്ഗിൽ എന്ത് നടക്കുന്നില്ല? ഫെർട്ടിലൈസേഷൻ നടക്കാതെ unfertilized egg develops into male. Unfertilized egg develops into male. Develops as a male, അല്ലെ. Drawn. ആ പ്രോസസ് നമ്മൾ എന്താ വിളിക്കുക? Without fertilization egg is developed into male, അല്ലെ. Male progeny. അതിനെ നമ്മൾ വിളിക്കുന്ന പേരാണ് parthenogenesis. That process is called parthenogenesis. Parthenogenesis. Without fertilization, the female egg develops into a male. അതാണ് നമ്മൾ പറഞ്ഞത്. Unfertilized egg develops as a male or drawn by means of parthenogenesis. അതിനെ നമ്മൾ വിളിക്കുന്ന പേരാണ് പാർത്തിനോ. അപ്പോൾ അവരുടെ ക്രോമസോം നമ്പർ എന്തായിരിക്കും? വിത്തൗട്ട്, അല്ലെ, വിത്തൗട്ട് ഫെർട്ടിലൈസേഷൻ the males are producing the haploid 16. It will be 16. അപ്പോൾ മെയിൽസിന്റെ ക്രോമസോം നമ്പർ എത്രയാ? n is equal to 16. Females it will be 2n is equal to 32. Females there are 32. Okay, this means that males have the half the number of chromosome than that of the female. Males in 16, females 32. 32. Males half the set, half the number of the chromosome that of a female. The females are diploid having 32 chromosome and males are haploid having 16 chromosomes. This is called haplodiploid sex determination. It is called diploid sex determination. Special type of sex determination is honeybees. Honeybees, wasp, ants. Honeybees, wasp, and ants. Okay. In another characteristic feature, such the males are 16 number, half the number of the chromosome. Males produce the gametes, male gametes, by the process of mitosis. By the process of mitosis. By the process of mitosis. Another important feature, males produce sperms by mitosis. And they do not have father and thus cannot have sons. Father and there will be not sons, but have grandfather, because that is the females that will produce by parental, both male and female gamete fused together. So there will be a grandfather, there will be there, and the grandsons there will be. The son will be grandson. Okay, but this is the sex determination in honeybee. About sex determination honeybee is haplodiploid. Here the chromosome number in female, it will be a diploid set of the chromosome. Males are having 16. Or males are produced in the female again without fertilization. The males are produced or drones are produced, and the chromosome number is half than the female. And male gametes produces male gametes. Males produce male gametes by the process of mitosis, and the chromosome number is half. Chromosome number is half. Okay, so that is the another sex determination in honeybees. Okay, then next we have to go through another feature, mutation. Next process is called mutation. Mutation is the phenomenon which results in the alteration of DNA sequences. So there is sudden and drastic irritable changes is called mutation. Sudden and drastic irritable changes, that is called mutation. That will result in the alteration or changes in DNA. And consequently, DNA sequence changes that will result the changes in the genotype and phenotype of an organism. Okay. In addition to recombination, mutation is another phenomenon that leads to variation in DNA. Well, variation in DNA that will also leads to mutation. And next, chapter number 6, one DNA helix runs continuously from one end to the other in each chromatid in a highly supercoiled form. Therefore, loss, deletion, or gain, insertion of a segment in DNA result in the alteration of the chromosome. But there will be loss of loss, or a part of a chromosome will lost, or there will be inserted or duplicated a segment of DNA that will result in the alteration in the chromosome. And that process, such that genes are located on the chromosome, that will be alteration in chromosome. Alteration in chromosome will also takes place. But genes chromosome is also lost. Chromosome part is also lost. That will result in the abnormalities or aberration. And that chromosomal aberrations are commonly observed in cancer cells. But what are the type of chromosomal aberrations? Deletions, loss or gain, insertion. A segment is lost, or segment is added or duplicated, or it may be inverted, inversion or translocation. There are 4 types of chromosomal aberrations. In addition to the above mutation, these are called chromosomal aberrations or chromosomal mutation, and it is mainly due to cancer, mainly the causative of cancer cells. Second, in addition to the above mutation, also arises due to change in a single base pair of DNA. Mutation arises in a single base pair of DNA. This is known as point mutation. Single base pair mutation, that is called point mutation. A classical example for mutation is sickle cell anemia. Sickle cell anemia you have already covered. That is in the case of here the sickle cell anemia, there will be a single point mutation. That is point, single gene mutation or single point mutation. Here a single base pair will be changing from adenine to uracil, GAG2. GAG codes for glutamic acid. GAG codes for valine. But the valine instead of glutamic acid, in the beta chain of globin molecule. Beta chain in the globin molecule. That will cause this point mutation. This mutation is called a point mutation. Single base pair of DNA. Single base pair of DNA. But GAG to GUG. Adenine is replaced by uracil. Adenine is replaced by uracil. That is called a single base pair mutation. Single base pair mutation. That is leads to sickle valine instead of glutamic acid in the 6th codon. 6th codon. No, last codon. Next disorders. 6th codon. 6th codon. Valine instead of glutamic acid. Glutamic acid valine. That will leads to that is called sickle cell. That RBC shape will be changed from the round and biconcave to elongated and sickle shape. Elongated and sickle shape. And the oxygen carrying capacity is decreases. That will leads to severe anemia. Severe anemia. That is sickle cell anemia. It is typical example for single pair of DNA. Change in the single pair. This is known as point mutation, or it is called a gene mutation. Point mutation or gene mutation. Typical example is sickle cell anemia. That's another mutation. Our chromosomal mutation, gene mutation, and deletions and insertions of base pairs of DNA causes frame shift mutation. That also seen in the chapter 6. That is molecular basis of inheritance. Next chapter that is molecular basis of inheritance. Frame shift mutation. That is called either deletion or insertion. That will change the reading frame. The reading frame will be changed. Reading frame will be changed. That is called frame shift mutation. That is a typical example in your book. It is given. RAM has red cap. RAM has red cap. Here it will be either an insertion of a letter here. Insertion of a letter here. Then the reading frame will be changed. RAM has after insertion DRE, DCA, P. Okay, the reading frame will be changed. Reading frame will be changed. If it is adding one more letter, BI. Okay, what is the reading frame? RAM has BIR, EDC, AP. Then one more letter is added, G. Then it will be, what is the segment? Next, the reading frame. RAM has big red cap. So the meaning will be changed. RAM has red cap, RAM has big red cap. The meaning will be changed. Here, there will be no importance of RAM has red or cap. It will be the genetic codons. They are the genetic codons. Genetic codons. And each codon will coding for an amino acid. Each codon will coding for an amino acid. But that the reading frame of this amino acid will be changed. That will affect the protein. That will affect the protein sequence. Protein sequence. That is called the frame shift mutation. That mutations are called frames. Our reading frame will change either deletion. Either deletion or insertion. So, insertion is delete. Here the deletion R is deleted. R is deleted. Then the reading frame will be changed from here. EDC AP. EDC AP. Okay, there will be no meaning. Then next E is again deleted. Then RAM has DCAP. DCAP. Then D is also deleted. Then the meaning will be again changed. RAM has cap. RAM has cap. Or RAM has red cap. It will be changed to RAM has cap. But the reading frame will be changed at the point of deletion. At the point of deletion or insertion. Point of deletion or insertion, the reading frame will be changed. And it is called frame shift mutation. Frame shift mutation. Okay. Next, the mechanism of mutation is beyond the scope of this discussion at this level. Or there are many chemical and physical factors that induce mutation. About the factors that will affect or induce mutation, they are called mutagens. They are called mutagen. The substances, chemical or physical factors that will induce mutation, they are called mutagens. Either the mutagens may be chemical mutagens or physical mutagens. UV radiation is an example for an physical mutagen, and it causes mutation in organism. It is a mutagen, physical mutagen. UV rays, gamma rays, X-rays. They are most of the ionizing radiation. Most of the ionizing radiations are physical mutagens, and some chemicals also. Mustard gas, sodium oxide, nitrous oxides. They are all chemical mutagens. The substance that cause mutation are called mutagens, either physical or chemical. Okay. Then next part that will be the gender disorders. Gender disorders. And we have to study in genetic order disorders. First one, pedigree analysis. First one is the pedigree analysis. And this is the main aim for this pedigree analysis. What is the main aim? Here in the case of the disorders that inherited has been prevailing in the human society since long. This was based on the heritability of certain characteristic features in families. അപ്പോൾ നമ്മൾ ഈ ഡിസോർഡേഴ്സ് ഒരു ഫാമിലിയിലൂടെ എങ്ങനെയാണ് പാസ് ചെയ്യുന്നത് എന്നുള്ളത് എന്തിനെ ബേസ് ചെയ്തിട്ടാണ് കണ്ടുപിടിക്കുന്നത്? ഹെറിറ്റബിലിറ്റി ഓഫ് സർട്ടൺ ക്യാരക്ടറിസ്റ്റിക് ഫീച്ചേഴ്സ് ഇൻ ഫാമിലി. അപ്പോൾ ഒരു ഫാമിലിയിൽ ഒരു ക്യാരക്ടർ ഇങ്ങനെ പാസ് ചെയ്ത് പോകുമ്പോൾ നമുക്ക് കണ്ടറിയാൻ പറ്റും. ഒരു ജനറേഷൻ അടുത്ത ജനറേഷനിലേക്ക്, അതിൽ നിന്ന് ജനറേഷനിൽ 2-3 ജനറേഷനിലേക്ക് നോക്കി കഴിഞ്ഞാൽ ഒരു ക്യാരക്ടർ ഇങ്ങനെ പാസ് ചെയ്ത് പോകുന്നത് കാണാൻ പറ്റും അല്ലേ? ആഫ്റ്റർ ദ റീഡിസ്കവറി ഓഫ് മെൻഡൽസ് work and the practice of analyzing the inheritance pattern of traits in human being. അപ്പോൾ ഹ്യൂമൻ ബീയിങ്സിൽ ആ പ്രോസസ്സ് സ്റ്റാർട്ട് ചെയ്ത് നോക്കി, അനലൈസിങ് ദ പ്രാക്ടീസ് ഇൻഹെറിറ്റൻസ് പാറ്റേൺ. പക്ഷേ അത് ഏതുപോലെ ചെയ്യാൻ പറ്റുന്നില്ല. സിൻസ് ഇറ്റ് ഈസ് എവിഡന്റ് ദാറ്റ് ദ കൺട്രോൾ പ്രോസസ് ദാറ്റ് ക്യാൻ ബി പെർഫോംഡ് ഇൻ പീ പ്ലാൻ അല്ലെ, മെന്റിൽ ഏതിലെ ചെയ്ത പീ പ്ലാന്റിൽ ചെയ്തപോലെ മനുഷ്യരിൽ ചെയ്യാൻ പറ്റുമോ? ഇല്ല. കൺട്രോൾ പ്രോസസ് ചെയ്യാൻ പറ്റുമോ? തിരിച്ച് ക്രോസ് ചെയ്യാൻ പറ്റുമോ? ഇല്ല. Some other ടെസ്റ്റ് ക്രോസ് കണ്ടക്ട് ചെയ്യാൻ പറ്റുമോ? ഇല്ല. അപ്പോൾ some other organism are not possible in case of the human beings. ഹ്യൂമൻ ബീയിങ്സിൽ ആ കേസ് ഒന്നും ചെയ്യാൻ പറ്റില്ല. നമുക്ക് അങ്ങനെയുള്ള ക്രോസുകളൊന്നും ചെയ്യാൻ പറ്റില്ല. അപ്പോൾ സ്റ്റഡി ഓഫ് ദ ഫാമിലി ഹിസ്റ്ററി അബൌട്ട് ഇൻഹെറിറ്റൻസ് ഓഫ് എ പർട്ടിക്കുലർ ട്രേറ്റ് പ്രൊവൈഡ്സ് ആൻ ഓൾട്ടർനേറ്റീവ്. ആകെ ഒരു ഓൾട്ടർനേറ്റീവ് ഉള്ളൂ. ഹ്യൂമൻ പെഡിഗ്രി ചാർട്ട് അല്ലെങ്കിൽ ഹ്യൂമൻ ഡിസോർഡേഴ്സ് പഠിക്കണമെങ്കിൽ നമ്മൾ എന്ത് ചെയ്യണം? ദാറ്റ് ഇസ് ഫാമിലി ഹിസ്റ്ററി പഠിക്കണം. ഫാമിലി ഹിസ്റ്ററി പഠിച്ചിട്ട് വേണം നമ്മൾ ഇൻഹെറിറ്റൻസ് ഓഫ് എ പർട്ടിക്കുലർ ട്രേറ്റ് ദാറ്റ് പ്രൊവൈഡ്സ് ആൻ ഓൾട്ടർനേറ്റീവ്. അപ്പോൾ ഫാമിലി ട്രേറ്റ്സ് പഠിക്കുക, അല്ലെങ്കിൽ ഒരു ഫാമിലി ട്രീയിലൂടെ പോകുന്ന ഒരു ഫാമിലി അല്ലെ, നമ്മളൊരു ഫാമിലി ട്രീ എന്ന് പറഞ്ഞു കഴിഞ്ഞാൽ നമ്മുടെ ഒരു 5-10 തലമുറ അങ്ങനെ വരുന്നത് നമ്മൾ നോക്കിയിട്ടാണ് ഒരു ഫാമിലി ട്രീയിലൂടെ ഒരു ക്യാരക്ടർ പോകുന്നത് നോക്കുന്നത്. സച്ച് ആൻ അനാലിസിസ് ഓഫ് ട്രേറ്റ്സ് or characters in a several of generation of a family is called pedigree analysis. അങ്ങനെ നമ്മൾ ഒരു ട്രേറ്റ് അല്ലെങ്കിൽ ഒരു ക്യാരക്ടർ അല്ലെങ്കിൽ ഒരു ഡിസോർഡർ ആവാം, ഒരു ഡിസോർഡർ അല്ലെ, ഒരു ഡിസോർഡർ ഒരു ജനറേഷനിൽ അടുത്ത ജനറേഷനിലേക്ക് ഡിസോർഡർ ട്രാൻസ്മിറ്റ് ചെയ്ത് പോയി അല്ലെ, അത് മുഴുവൻ അനലൈസ് ചെയ്യണമെങ്കിൽ നമുക്ക് എന്ത് വെച്ചിട്ട് ചെയ്യാൻ പറ്റുള്ളൂ? പെഡിഗ്രി അനാലിസിസ് വെച്ചിട്ട് ചെയ്യാൻ പറ്റുള്ളൂ. In the pedigree analysis, the inheritance of a particular trait is represented in the family tree over generations. Inheritance of a particular trait represents the family tree over generation. അതുകൊണ്ടാണ് ഹ്യൂമൻ ജെനറ്റിക്സിലെ പെഡിഗ്രി സ്റ്റഡി പ്രൊവൈഡ്സ് എ സ്ട്രോങ് ടൂൾ വിച്ച് ഈസ് യൂട്ടിലൈസ്ഡ് ടു ട്രേസ് ദ ഇൻഹെറിറ്റൻസ് ഓഫ് എ സ്പെസിഫിക് ട്രേറ്റ്, അബ്നോർമാലിറ്റി ഓർ ഡിസീസ്. അപ്പൊ നമുക്കൊരു അബ്നോർമാലിറ്റി അല്ലെങ്കിൽ ഡിസീസോ അല്ലെങ്കിൽ ഒരു ട്രേറ്റോ ഒരു ഫാമിലി ട്രീയിലൂടെ എങ്ങനെയാണ് പാസ് ചെയ്യുന്നത് എന്നുള്ളത് അല്ലെ, അതാണ് പെഡിഗ്രി സ്റ്റഡിയിലൂടെ നമുക്ക് കിട്ടുന്നത്. ഇനി അതിന് നമ്മൾ പെഡിഗ്രി സ്റ്റഡീസ് പഠിക്കുമ്പോൾ അതിന് ഇമ്പോർട്ടൻ്റ് ആയിട്ട് വേണ്ടത് സം ഓഫ് ദി ഇമ്പോർട്ടൻറ്റ് സ്റ്റാൻഡേർഡ് സിമ്പിൾസ് യൂസ് ചെയ്യുന്നു പെഡിഗ്രി അനാലിസിസിൽ. ആ സിമ്പിൾസ് ആണ് നമ്മൾ കണ്ടത്. സ്ക്വയേഴ്സ് ഫോർ മെയിൽസ്, സർക്കിൾസ് ഫോർ ഫീമെയിൽസ്, ആൻഡ് ദീസ് റോംബോയിഡ് സ്ട്രക്ചർ ഫോർ സെക്സ് അൺസ്പെസിഫൈഡ്, ആൻഡ് സോളിഡ് സിമ്പിൾസ് ആർ അഫക്റ്റഡ് ഇൻഡിവിജ്വൽസ്, ആൻഡ് ദർ ഇസ് എ മേറ്റിംഗ് ലൈൻ ഓർ മാരേജ് ലൈൻ, the male and female, mating line or marriage line. Then this is the mating between relatives, marriage between relatives ആണ് നമ്മൾ രണ്ട് horizontal line ഒഴിക്കുന്നത്. രണ്ട് horizontal line വെച്ച് കഴിഞ്ഞാൽ that is called mating between relatives. ഇതിനെ നമ്മൾ വിളിക്കുന്ന പേരാണ് consanguineous mating. Mating between relatives നമ്മൾ വിളിക്കുന്ന പേരാണ് consanguineous mating. അപ്പോൾ അവിടെ നമ്മൾ രണ്ട് horizontal line വെച്ചിട്ടാണ് കണക്ട് ചെയ്യുക. ഇനി അടുത്ത ഒരു ജനറേഷൻ കഴിഞ്ഞ് അടുത്ത ജനറേഷനിലേക്ക് അല്ലെ കുട്ടികൾ വരുമ്പോൾ നമ്മൾ എങ്ങനെ കാണിക്കുക? പാരന്റ്സ് അബോവ് ആൻഡ് ചിൽഡ്രൻ ബിലോ അല്ലെ, ചിൽഡ്രൻ. അപ്പൊ ഒരു വെർട്ടിക്കൽ ലൈൻ ഇട്ടിട്ടാണ്, ഹൊറിസോണ്ടൽ ലൈനിൽ നിന്ന് ഒരു വെർട്ടിക്കൽ ലൈൻ കൊടുത്തിട്ടാണ് നമ്മൾ എന്ത് കൊടുക്കുന്നത്? ചിൽഡ്രൻ. അത് എവിടുന്ന? ഓർഡർ ഫ്രം ബർത്ത് അല്ലെ, ഓർഡർ ഓഫ് ബർത്ത് എവിടുന്ന കൊടുക്കുക? ലെഫ്റ്റ് ടു റൈറ്റ്. ദിസ് ഇസ് ഫസ്റ്റ് ചൈൽഡ്, ദിസ് സെക്കൻഡ് ചൈൽഡ്, ദെൻ സോ ഓൺ. ഫസ്റ്റ് സെക്കൻഡ്. അപ്പൊ ഫ്രം ലെഫ്റ്റ് ടു റൈറ്റ്. പെഡിഗ്രി സിമ്പിൾസ് ഒന്ന് ഐഡന്റിഫൈ ചെയ്ത് വെക്കുക. Then parents with the male child affected with the disease, then 5 unaffected offsprings. But that is some pedigree symbols it is given. There are 60 more pedigree symbols. 60 more pedigree symbols ഉണ്ട്. നമുക്ക് ഇമ്പോർട്ടൻറ്റ് സിമ്പിൾസ് നമുക്ക് ഇത് മാത്രമാണ് നമുക്ക് ടെക്സ്റ്റിൽ NCERT ടെക്സ്റ്റിൽ പഠിപ്പിക്കുന്നത്. മെയില്, ഫീമെയില് അല്ലെ, സെക്സ് അൺസ്പെസിഫൈഡ് ഓർ അൺഐഡന്റിഫൈഡ് സെക്സ്, then affected means what? Diseased individuals, affected individuals or diseased individuals are solid symbols or shaded symbols. Okay, then mating line, mating between close relatives, then first generation mark ചെയ്യുമ്പോൾ ശ്രദ്ധിക്കണം അല്ലേ. That is the case of, this is called first generation. Roman letters കൊടുക്കുക. This is the second generation. Roman letters ആണ് നമ്മൾ ഫസ്റ്റ് ജനറേഷൻ, സെക്കൻഡ് ജനറേഷൻ, അങ്ങനെ ജനറേഷൻ നമ്മൾ റോമൻ ലെറ്റേഴ്സ്. അതിന് ഒരു ജനറേഷനിൽ തന്നെ ഇൻഡിവിജ്വൽസിനെ അല്ലെ, ഫസ്റ്റ് ജനറേഷനിൽ ഇൻഡിവിജ്വൽസിന് നമ്മൾ മാർക്ക് ചെയ്യുക അറബ് ന്യൂമറൽസ്. Arab numerals ആണ് ഉപയോഗിക്കുക 1 അല്ലേ. അപ്പോൾ ഫസ്റ്റ് ജനറേഷനിൽ ആദ്യത്തെ ഇൻഡിവിജ്വൽ, സെക്കൻഡ് ഇൻഡിവിജ്വൽ അല്ലേ, സെക്കൻഡ് ജനറേഷനിൽ ഫസ്റ്റ് ഇൻഡിവിജ്വൽ, സെക്കൻഡ് ഇൻഡിവിജ്വൽ, അങ്ങനെയാണ് നമ്മൾ അതിനെ ലാബിൽ ചെയ്യുക. ദാറ്റ് ഇസ് ദ ലാബിലിംഗ്. അത് ബുക്കിൽ പറയുന്നില്ല, പക്ഷെ അത് നമ്മൾ അറിഞ്ഞു വെക്കണം. ജനറേഷൻ 1 2 3 4 എക്സെട്ര അത് റോമൻ ലെറ്ററിൽ കൊടുക്കും. അല്ല ഇൻഡിവിജ്വൽസിന് നമ്മൾ കൊടുക്കുന്നത് ഓരോ ജനറേഷനിൽ വരുന്ന ഇൻഡിവിജ്വൽസിനെ 1 2 അല്ലെ അറബ് ന്യൂമറൽസ് ഇട്ടാണ് കൊടുക്കുക. അപ്പൊ അങ്ങനെ കുറച്ച് സിമ്പിൾസ് നമ്മൾ പഠിച്ചു വയ്ക്കുന്നത് നല്ലതാണ്. ദാറ്റ് ഇസ് വെരി ഇമ്പോർട്ടൻറ്റ് ഫോർ പെഡിഗ്രി അനാലിസിസ്. അത് ഇതുപോലെ 60 സിമ്പിൾസ് ആണ്, പക്ഷെ നമുക്ക് NCERT ടെക്സ്റ്റിൽ നമുക്ക് ഇതിന്റെ അപ്ലിക്കേഷനേ ഉള്ളൂ. 60 ഒന്നും നമുക്ക് ആവശ്യം വരുന്നില്ല. ഓക്കെ, ഇനി നമ്മൾ ലാസ്റ്റ് ഇൻ ഹ്യൂമൻ ജെനറ്റിക്സ് പെഡിഗ്രി പ്രൊവൈഡ്സ് strong tool which is utilized to trace the inheritance of a specific trait, abnormality or disease, and some of the important symbols are also used in the pedigree analysis shown in the figure 5.13. Then you have also studied this chapter. Each and every feature in any organism is controlled by one or the other gene located on the DNA present in the chromosome. Okay, അപ്പോൾ നമ്മൾ പഠിച്ചു കഴിഞ്ഞ ഈ ചാപ്റ്ററിന്റെ തുടക്കത്തിൽ തന്നെ നമ്മൾ പഠിച്ചു ഈച്ച് ആൻഡ് എവരി ഫീച്ചർ മീൻസ് വാട്ട് ക്യാരക്ടർ in any organism is controlled by one or the other gene located on the DNA, and it will be present in the chromosome. Okay, then DNA is the carrier of the genetic information. DNA is the carrier of genetic information, and it is hence transmitted from one generation to the other without any change or alteration. Normally that characters will be transmitted without any change, without any change or alteration, hence transmitted from one generation to the other without any change. However, there will be some changes takes place in DNA, changes or alteration to takes place occasionally. Changes in such an alteration or change is called mutation. That changes are called mutation. Then a number of disorders in human beings have found to be associated with the inheritance of changed or altered genes or chromosome. അതാണ് നമ്മൾ നെക്സ്റ്റ് കാണാൻ പോകുന്നത്. നമ്പർ ഓഫ് ഡിസോർഡേഴ്സ്, ചേഞ്ചസ് ഓർ മ്യൂട്ടേഷൻ അല്ലെ, ദാറ്റ് വിൽ ഫോം സം ഡിസോർഡേഴ്സ് ദാറ്റ് വിൽ ബി ട്രാൻസ്ഫേർഡ് ടു ഫ്രം ജനറേഷൻ ടു ജനറേഷൻ ഹാവ് ബീൻ ഫൗണ്ട് ടു ബി അസോസിയേറ്റഡ് വിത്ത് ഇൻഹെറിറ്റൻസ് ഓഫ് ചേഞ്ച്ഡ് ഓർ ആൾട്ടേർഡ് ജീൻസ് ഓർ ക്രോമസോം. മെന്റലി അതിന് നമ്മൾ ഡിസോർഡേഴ്സിന് നമ്മളൊന്ന് ക്ലാസിഫൈ ചെയ്യാൻ പോവാണ്. ഫസ്റ്റ് ബ്രോഡ്ലി, അപ്പൊ അതിന്റെ ഹെഡിങ് നമ്മൾ മെന്റേലിയൻ ഡിസോർഡർ എന്നാണ് കൊടുത്തിട്ടുള്ളത്. ഹെഡിങ് മെന്റേലിയൻ ഡിസോർഡർ. ബ്രോഡ്ലി genetic disorders may be grouped into two categories. What are the categories? One is called Mendelian disorder and the other is called chromosomal disorder. Mendelian disorders are mainly due to the disorder that will take place by alteration or mutation in a single gene. Alteration or mutation in a single gene. That is very important point. That is the case of Mendelian disorder. But there are two disorders, Mendelian disorders and chromosomal disorders. And Mendelian disorders are mainly due to the alteration or mutation in the single gene. These single gene mutations are called Mendelian disorders. And these disorders are transmitted to the offspring on the same line as we have studied in the principles of inheritance or Mendelian inheritance. These disorders are transmitted to the offspring on the same lines as we have studied in the principles of inheritance, and it is called Mendelian disorders. Mendelian disorders. Okay. The pattern of inheritance of such Mendelian disorders can be traced in a family by the pedigree analysis. That is very important point. The pattern of inheritance of such Mendelian disorders. Mendelian disorders എങ്ങനെയാണ് ഒരു ഫാമിലി ട്രീയിലൂടെ പോകുന്നത് നമുക്ക് എങ്ങനെ ട്രേസ് ചെയ്യാം? പെഡിഗ്രി അനാലിസിസ് ഉപയോഗിച്ച് ട്രേസ് ചെയ്യാം. പെഡിഗ്രി അനാലിസിസ് ഉപയോഗിച്ച് നമുക്ക് മെന്റേലിയൻ ഡിസോർഡേഴ്സ് ഒരു ഫാമിലിയിലൂടെ എങ്ങനെയാണ് പാസ് ചെയ്യുന്നത് അതിന്റെ പാറ്റേൺ നമുക്ക് കണ്ടുപിടിക്കാൻ പറ്റും എന്ന് പറയുന്നത്. ഓക്കെ, ഇനി കുറച്ച് പ്രിവലന്റ് ആയിട്ടുള്ള മോസ്റ്റ് കോമൺ ആൻഡ് പ്രിവലന്റ് മെന്റേലിയൻ ഡിസോർഡേഴ്സിന്റെ പേരാണ് ഹീമോഫീലിയ, സിസ്റ്റിക് ഫൈബ്രോസിസ്, sickle cell anemia, color blindness, phenylketonuria, and thalassemia. There are the 6 or 7. It will be a 6. Hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, and thalassemia. There are 6 Mendelian disorders. And it is important to mention that here such Mendelian disorders may be dominant or recessive. That is, go through this dominant alleles or recessive alleles. By pedigree analysis, one can easily understand whether the trait in question is either dominant or recessive. നമുക്ക് ഒരു പെഡിഗ്രി അനാലിസിസിലൂടെ നമുക്ക് എന്ത് ചെയ്യാൻ പറ്റും? ഒരു ട്രേറ്റ് ഡോമിനന്റ് ആണോ റിസസീവ് ആണോ? അതാണ് നമ്മൾ അടുത്ത രണ്ട് പഠിക്കാൻ പോകുന്നത്. This is the dominant disorder. Go through this pedigree analysis, and this one is the recessive. Recessive disorder, dominant and recessive. And it may be either autosomal. This is autosomal dominant and autosomal recessive. Autosomal ano ado similarly the trait may also be linked to the sex chromosome. About autosome ano ado sex chromosome ano, as in the case of hemophilia. Hemophilia is a sex linked disorder ആണ് ഹീമോഫീലിയ. ഇത് നമ്മൾ കാണുന്ന ഓട്ടോസോമൽ, ഇപ്പോൾ സിക്കിൾ സെൽ അനീമിയ, ദെൻ മയോട്രോണിക് ഡിസ്ട്രോഫി, ഇതെല്ലാം നമുക്ക് കാണുന്ന ഓട്ടോസോമൽ ഡിസോർഡേഴ്സ് ആണ്. അപ്പോൾ ദാറ്റ് ഡിസോർഡേഴ്സ് മേ ബി ഐദർ ഡോമിനന്റ് ഓർ റിസസീവ്, ഓർ ഇറ്റ് മേ ബി ഓട്ടോസോമൽ ഓർ സെക്സ് ക്രോമസോമൽ. ഓട്ടോസോം ഓർ സെക്സ് ക്രോമസോം. ഓട്ടോസോം ഓർ സെക്സ് ക്രോമസോം. As in the case of hemophilia. Sex chromosome വരുന്ന disorders ആണ്, അല്ലെങ്കിൽ sex linked disorders ആണ് hemophilia, and one more color blindness. Color blindness. അപ്പോൾ അത് നമുക്ക് typical examples നമുക്ക് പഠിക്കാനുള്ളതാണ്. Okay. Then it is evident that in the case of hemophilia, ഹീമോഫീലിയയിൽ നമ്മൾ നോക്കി കഴിഞ്ഞാൽ it is evident that this X-linked recessive, ഇത് സെക്സ് ക്രോമസോമിലാണ്, അത് ഏത് ക്രോമസോമിലാണ് സെക്സിലെ X-linked recessive trait shows the transmission from carrier female to male progeny. Carrier female എന്ന് പറയുമ്പോൾ നമ്മള് X അല്ലെ, സ്മോൾ h, carrier. Normal gene, normal allele is the recessive, that is the case of the disease causing അല്ലെ, hemophilic allele. ഇതാണ് carriers. Carrier female, and she has no symptom of hemophilia അല്ലെ. Carrier female to male progeny. അപ്പോൾ ഈ ഫീമെയിൽ കാരിയർ ക്രോസ് വിത്ത് മെയിൽ അല്ലെ, മെയിലും കൂടി മാരേജ് ആകുമ്പോൾ നമുക്ക് കാണാൻ പറ്റും. ദാറ്റ് ഇസ് ദ കേസ് ഓഫ് ഇറ്റ് വിൽ പ്രൊഡ്യൂസ് സം മെയിൽ. ഇപ്പോൾ ഫീമെയിൽസ് എടുക്കുമ്പോൾ XX ആൻഡ് X അല്ലെ, സ്മോൾ h X. ഓക്കെ, ദെൻ Y ക്രോമസോം മെയിൽസ് എടുക്കുമ്പോൾ XY നോർമൽ. അടുത്തത് this X and this Y, that is X small h Y. X small h Y, that will be hemophilic. It will be hemophilic. But here this X-linked resultate shows the transmission from carrier female to male progeny. ഇതാണ് നമ്മൾ കാണുന്നത്. Okay, then a representative pedigree shown figure for dominant and recessive traits are given below. Dominant pedigree chart and recessive pedigree discuss teacher. Okay, അപ്പോൾ ഇത്രയും കാര്യങ്ങൾ നമുക്ക് ഈ ചാപ്റ്ററിൽ നമുക്ക് നോക്കാനുള്ളത് നെക്സ്റ്റ് പെഡിഗ്രി അനാലിസിസ് ആണ്. നമ്മൾ പറഞ്ഞ അതിലൊന്ന് ഇത് ഓട്ടോസോമൽ ഡോമിനന്റ് ആണ്, മയോട്രോണിക് ഡിസ്ട്രോഫി. ദിസ് വൺ ഈസ് ഓട്ടോസോമൽ റിസസീവ്, ദാറ്റ് ഈസ് സിക്കിൾ സെൽ അനീമിയ. ഓക്കെ, അപ്പോൾ ഇവിടെ നമുക്ക് ഡോമിനന്റ് അലീൽ എന്ന് പറയുമ്പോൾ ഇവിടെ നമുക്കറിയാം ക്യാപിറ്റൽ എ ഓർ ക്യാപിറ്റൽ എ സ്മോൾ എ. പക്ഷെ ഇവിടെ എന്ത് വരുന്നുണ്ട്? സ്മോൾ എ വരുന്നുണ്ട്. ദീസ് ആർ unaffected അല്ലെ, unaffected small a. അപ്പോൾ ഹിയർ ഇറ്റ് വിൽ ബി ക്യാപിറ്റൽ എ സ്മോൾ എ. അതുകൊണ്ടാണ് ഇവിടെ എന്ത് വരുന്നത്? സ്മോൾ എ അല്ലെ. ഹിയർ ഓൾസോ സ്മോൾ എ, then here small a, here capital A small a, here also capital A small a. And this male marries another female, that is normal, without that is in the case of normal female without unaffected. Then all of them are small a with small a, all of them are unaffected. Okay, then here it will be capital A small a, here it will be small a male, marriage with the small a. Then it will be small a, capital A small a, and small a. That is in the case of this autosomal dominant disorder. അതുപോലെ ഇത് നമ്മൾ ഓട്ടോസോമൽ റിസസീവ് ഡിസോർഡർ ആണ്. ഇവിടെ നമുക്കറിയാം ദാറ്റ് ഇസ് ഇൻ ദ കേസ് ഓഫ് ഹിയർ സിക്കിൾ സെൽ അനീമിയ അല്ലെ. സിക്കിൾ സെൽ അനീമിയയിൽ നമുക്കറിയാം ഹിയർ ഇറ്റ് വിൽ ബി HBS സിക്കിൾ സെൽ, HBS അല്ലെ, സിക്കിൾ സെൽ. അപ്പൊ അങ്ങനെ വരണമെങ്കിൽ ഇവിടെ എന്തായിരിക്കണം? both the parents are carriers. Both the parents are heterozygous or carriers. Then HBS. Here also, both these here it will be carriers. HBS. Carriers are having no sickle cell anemia. That not affected. And here the progeny will be affected. That will be HBS. For 50% it will be the allele for sickle cell, and 50% will be normal allele. So both the parents are carriers in the case of sickle cell anemia, or it is a autosomal recessive trait, and here this will be autosomal dominant trait. Autosomal dominant trait. Okay, the next one is color blindness. Color blindness. It is a sex-linked recessive disorder due to the defect in either red or green cone of eye, resulting in the failure of discriminating between red and green color. Color blindness is a sex-linked recessive disorder due to the effect of either red or green cone of the eye, resulting in the failure of discriminate between red and green color. Red and green color. And this defect is due to mutation in certain genes present on the X chromosome. X chromosome. And it occurs about 8% of males and only about 0.4% in females. Most of this color blindness is occurred in males because males are having only one X chromosome. Females have two X chromosome and the percentage will be very less, 0.4%. Here most severe in the case of males because there are only one X chromosome. But this is because the genes that lead to the red-green color blindness are on the X chromosome. And males have only one X chromosome and females have two X chromosome. Males have one X chromosome and females have two X chromosome. About the son of a woman who carries the gene has a 50% chance of being colorblind. The sons of a woman who carries the gene has a 50% chance of being colorblind. The mother is not herself colorblind because the gene is recessive. That is X-linked recessive. That means its effect is suppressed by matching dominant normal gene. Now, females and X. Here it will be colorblindness, small c. But that is in the case of this one, the 50% of a mother have the recessive allele and the other 50% having suppressed by the dominant matching allele. Dominant matching dominant normal gene. But that will be masked. Here it is in the case of females, the effect will be only 0.4%. Whereas males have only one X chromosome. But the chance will be about 8% of males are having color blindness. That is X-linked recessive disorder. The mother is not colorblind because the gene is recessive. That means that its effect is suppressed by her matching dominant normal gene. And the daughter will not normally be colorblind. Marriage normal XY. Marriage the daughters are normally either normal ones or carriers. Normal ones or carriers. There will be no colorblind. Normal ones or colorblind. Unless if the daughter will be a colorblind, a daughter will not normally be colorblind. Unless daughter colorblind, her mother is the carrier and father is colorblind. Mother is carrier and father is colorblind. Father colorblind, the daughter is also got X small c. That got the color blindness. That is very rare case. Then next, hemophilia. Artha sex-linked or X-linked disorder, hemophilia, like color blindness. This is also the sex-linked recessive disease or X-linked, which shows its transmission from unaffected carrier female to some of the male progeny. Some of the male progeny has been widely studied. X small h with then there will be chance will be X small h y. That is the case of 50% of the males are affected. But that is transmission from unaffected carrier female to some of the male progeny. In this disease, a single protein, what is the causative of this hemophilia? Single protein that is a part of the cascade of proteins, steps-wise, cascade, steps of protein, stepwise reaction, step of proteins involved in the clotting of blood is affected. Due to this, an affected individual as a simple cut will result in non-stop bleeding. Non-stop bleeding. An affected individual, a simple cut will result in non-stop bleeding. The heterozygous female carrier for hemophilia may transmit the disease to sons. About female to sons. Female sons. Okay. The possibility of a female becoming hemophilic is extremely rare. Color blindness. Female becoming hemophilic. Mother should be at least a carrier and father should be hemophilic. The family pedigree Queen Victoria shows a number of hemophilic descendants. She was a carrier of the disease. The Queen Victoria is a carrier of the disease. That's why a royal disease. Hemophilia is called a royal disease. Next one, sickle cell anemia. Another disorder is called sickle cell anemia. This is an also autosomal linked recessive trait that can be transmitted from parents to offspring when both the partners are carrier for the gene. Both the partners are carriers, HBA, HBS, or they are called heterozygous, carriers or heterozygous. The disease is controlled by a single file HBA and HBS. Out of the 3 possible genotypes, only homozygous individuals for HBS, that is called HBS, HBS, show the disease the phenotype. അപ്പോൾ നമ്മൾ 3 genotypes ആണ് ആ population normal ones, HBA, HBA. Then next carriers, HBA, HBS. Then sickle cell, HBS, HBS. Sickle cell. അപ്പോൾ അതാണ് നമ്മൾ പറഞ്ഞത്, that is the diseased phenotype. That is heterozygous HBA, HBS individuals appear apparently unaffected. ഇവർക്ക് എന്തില്ല sickle cell anemia, but they are carrier of the disease, and there is 50% probability of transmission of the mutant gene. 50% HBS, 50% mutant gene to the progeny, thus exhibiting sickle cell anemia. And the defect is caused by the reason. The defect is caused by the substitution of glutamic acid. Substitution of glutamic acid. Substitution of glutamic acid. Glutamic acid മാറിയിട്ട് ഇവിടെ ആര് വരാണ്? Valine. Glutamic acid by valine. അല്ലേ? എന്താ എത്രാമത്തെ പൊസിഷനിലാ? 6th position. 6th codon of the beta chain of the globin molecule. അപ്പോൾ glutamic acid by valine. അപ്പോൾ 6th ഇവിടെ glutamic acid ആയിരുന്നു. അപ്പോൾ ഇവിടെ GAG അല്ലേ? GAG codes for glutamic acid അല്ലേ? That will change to GUG. A will be replaced by U. അപ്പോൾ അവിടെ എന്തായി? glutamic acid പകരം valine. In the 6th codon of the beta chain of the globin molecule. അതാണ് നമ്മൾ hemoglobin molecule നമ്മൾ കാണുന്നത്. And the substitution of amino acid in the globin chain protein result due to a single base substitution at the 6th codon of the beta globin from GAG to GUG. ഞാൻ കാണിച്ചതാണ് GAG to GUG. Okay. The mutant hemoglobin molecule undergoes polymerization under low oxygen tension, causing the change in the shape of RBC from ബൈകോൺകേവ് ഡിസ്ക് ടു ഇലോങ്ങേറ്റഡ് സിക്ക്. അപ്പൊ ബൈകോൺകേവ് ഷേപ്പ് മാറിയിട്ട് ഏത് ഷേപ്പിലേക്ക് മാറി? സിക്കിൾ ഷേപ്പിലേക്ക് മാറി. അതാണ് നമ്മളോട് ഷേപ്പ് കണ്ടത്. ഓക്കെ, ഇതാണ് റൗണ്ട് ആൻഡ് ബൈകോൺകേവ്, ബോത്ത് ഇലോങ്ങേറ്റഡ് ആൻഡ് സിക്കിൾ ഷേപ്പ്. ഇറ്റ് വിൽ ബി സിക്കിൾ ഷേപ്ഡ്, ഇലോങ്ങേറ്റഡ്. അപ്പൊ ദ ഓക്സിജൻ ക്യാരിങ് കപ്പാസിറ്റി ഡിക്രീസസ് and it will form severe anemia. അല്ലെ, symptom will be severe anemia. Severe anemic ആയിരിക്കും. Okay, അതാണ് that is called that is sickle cell anemia, and it is connected with the autosomal recessive disorder, and chromosome number is 11. The next one is phenylketonuria. This inborn error of metabolism is also inherited as autosomal recessive trait. ഇത് നമ്മൾ പഠിച്ചു കഴിഞ്ഞാണ്. phenylketonuria എവിടെയാ? പ്ലിയോട്രോപിസിന് എക്സാമ്പിൾ പഠിച്ചാണ്. The affected individual lacks an enzyme that converts amino acid phenylalanine to tyrosine. അപ്പോൾ amino acid phenylalanine tyrosine ആക്കി മാറ്റുന്ന enzyme, അതിന്റെ പേരാണ് phenylalanine hydroxylase. That result enzyme will be lacking due to the gene mutation. That is lacks an enzyme that converts the amino acid phenylalanine to tyrosine. അതിന്റെ കാരണം എന്തുകൊണ്ടാണ്? That will be a mutation takes place in that gene. As a result of this, phenylalanine is accumulated and converted into phenylpyruvic acid and other derivatives. And accumulation of these in brain result in mental retardation. This will leads to mental retardation, and these are excreted through urine because of its pure absorption by kidney. These are also excreted through urine because of its pure absorption. It is an inborn error of metabolism, and it is an autosomal recessive trait. And it is connected with the chromosome number 12. Phenylketonuria is connected with the chromosome number 12. The next disorder is called thalassemia. Thalassemia is also an autosomal linked recessive disorder, and it is also blood disease. Blood disease തന്നെയാണ്. ഏതുപോലെ തന്നെയാ? sickle cell anemia. Sickle cell anemia is a blood disease, and thalassemia is also a blood disease. And it is transmitted from parents to the offspring when both the partners are unaffected carrier. Both the partners are unaffected carrier for the gene, or it is called a heterozygous. Same as in the case of sickle cell anemia. Then the defect could be due to either mutation or deletion, which ultimately results in the reduced rate of synthesis of one of the globin chains. Globin chains number reduce ചെയ്യുമ്പോഴാണ് ഈ സിംറ്റം വരുന്നത്. ഏതാ? തലസീമിയ. അത് ഏതൊക്കെ ചെയിൻസ് ആണ്? ഹിമോഗ്ലോബിനും അറിയാം, ആൽഫ ചെയിൻ ആൻഡ് ബീറ്റ ചെയിൻ. ഒരു ഹിമോഗ്ലോബിൻ മോളിക്യൂളിൽ 2 ആൽഫ ചെയിനും 2 ബീറ്റ ചെയിനും ആണ്. ആൽഫ 1, ആൽഫ 2, ബീറ്റ 1 ആൻഡ് ബീറ്റ 2 അല്ലെ, 4 ചെയിനാണ് ഒരു ഹിമോഗ്ലോബിൻ മോളിക്യൂൾ. and it is made up of this quaternary structure of the protein. Okay, this causes the formation of abnormal hemoglobin molecule resulting into anemia, which is characteristic of the disease. But that is the number of the globin chain. Globin chain എണ്ണം കുറഞ്ഞു കഴിഞ്ഞാൽ alpha chain or beta chain, reduced rate of synthesis of one of the globin chain, alpha chain or beta chain, that will causing the symptom that is called anemic, anemia, abnormal hemoglobin molecules. അത് നമ്മൾ തലസീമിയനെ രണ്ടായിട്ട് ക്ലാസിഫൈ ചെയ്യാൻ പോവാണ്. അക്കോർഡിംഗ് ടു which chain of the hemoglobin molecule is affected. തലസീമിയയിൽ നമ്മൾ പറഞ്ഞു രണ്ട് ഹീമോഗ്ലോബിനിൽ രണ്ട് ചെയിൻ ഉണ്ട്, ആൽഫ ചെയിൻ ഉണ്ട്, ബീറ്റ ചെയിൻ ഉണ്ട്. ആൽഫ ചെയിനെ അഫക്ട് ചെയ്ത് കഴിഞ്ഞാൽ ആൽഫ ചെയിനെ അഫക്ട് ചെയ്ത് കഴിഞ്ഞാൽ ആ തലസീമിയ നമ്മൾ വിളിക്കുന്ന പേരാണ് ആൽഫ തലസീമിയ. The production of alpha globin chain is affected, while in beta thalassemia the production of beta globin chain is affected. Alpha thalassemia alpha globin chain affect ചെയ്യും, beta thalassemia the production of beta globin chain is affected. And alpha thalassemia is controlled by genes ന്റെ പേര് പഠിച്ചിരിക്കണം. Two closely linked genes HbA1 and HbA2. Alpha thalassemia is controlled by two closely linked genes HbA1 and HbA2 on chromosome number 16 of each parent. Chromosome number 16 of each parent, HbA1 and HbA2, രണ്ട് ജീൻസ് ആണ്, അത് രണ്ട് each parents അത് ക്രോമസോം നമ്പർ ഏതിലാണ് നിൽക്കുന്ന ജീൻസ് 16ലാണ് നിൽക്കുന്നത്. Okay, and it is observed due to the mutation or deletion of one or more of the four genes. രണ്ട് ജീന് ഒരു ക്രോമസോം, രണ്ട് ജീനകത്ത് ക്രോമസോമിൽ ആ ജീനുകളിൽ ഏതെങ്കിലും ഒന്നിൽ ചേഞ്ച് വന്നു കഴിഞ്ഞാൽ the more genes affected, the less alpha globin molecules are produced. Alpha globin molecules are produced. അപ്പോൾ the more genes are affected. ജീൻസ് ഏതൊക്കെ ജീൻസ് അഫക്ട് ചെയ്യുന്നത് ഈ 4 ജീൻസിൽ one or more കൂടുതൽ അഫക്ട് ചെയ്യുന്നവരും കുറയും ലെസ് ആൽഫ ഗ്ലോബിൻ ചെയിൻ മോളിക്യൂൾസ്. അപ്പോൾ ഹിമോഗ്ലോബിന്റെ പ്രൊഡക്ഷൻ കുറയും ഹിമോഗ്ലോബിൻ. Then while beta thalassemia is controlled by a single gene HBB on chromosome number 11. അപ്പോൾ beta thalassemia is controlled by the single gene HBB on chromosome number 11. ഇവിടെ 16ലാണെങ്കിൽ ഇവിടെ 11ല. alpha thalassemia 16ലാണെങ്കിൽ ഇവിടെ എത്രയാണ് 11 of each parent and occurs due to mutation of one or both the genes. അപ്പോൾ ജീൻസിൽ ചേഞ്ച് വന്നു കഴിഞ്ഞാൽ ആ ജീൻസിൽ ചേഞ്ച് വന്നതിൽ മ്യൂട്ടേഷൻ നടന്നു കഴിഞ്ഞാൽ ആ ബീറ്റ ഗ്ലോബിൻ ചെയിന്റെ നമ്പർ കുറയും. ബീറ്റ ഗ്ലോബിൻ റെഡ്യൂസിംഗ് ദ നമ്പർ ഓഫ് ബീറ്റ ഗ്ലോബിൻ. അതാണ് ദാറ്റ് ഇസ് ആൽഫ തലസീമിയ ആൻഡ് ബീറ്റ തലസീമിയ. ഇനി തലസീമിയ ഈസ് ഡിഫേഴ്സ് ഫ്രം സിക്കിൾ സെൽ അനീമിയ. ഫോർമർ ഇസ് എ ക്വാണ്ടിറ്റേറ്റീവ് പ്രോബ്ലം. എന്ന് വെച്ചാൽ തലസീമിയ ഇസ് എ quantitative problem because the globin chain, number of the globin chain will be affected in the case of thalassemia, while in the case of second one, that is later few globin molecules, while the later is a qualitative problem, that is sickle cell anemia, synthesizing and incorrectly functioning globin. Incorrectly functioning globin molecule structure change varana, മറ്റ് നമ്പറിൽ ചേഞ്ച് വരാണ്. ഇവിടെ ഗ്ലോബിൻ മോളിക്യൂളിന്റെ സ്ട്രക്ചറിൽ സിക്കിൾ സെൽ ഷേപ്പ് ആണ്, ഇറ്റ് വിൽ ബി സിക്കിൾ സെൽ. മറ്റ് നമ്പർ ഓഫ് ഗ്ലോബിൻ മോളിക്യൂൾ വിൽ ബി അഫക്റ്റഡ്. അപ്പോൾ ദാറ്റ് വിൽ ബി തലസെമിയ ഈസ് എ ക്വാണ്ടിറ്റേറ്റീവ് ആൻഡ് സിക്കിൾ സെൽ അനീമിയ ഈസ് ക്വാളിറ്റേറ്റീവ്. അതൊരു എക്സാമിന് ചോദിച്ചതാണ്, നീറ്റ് എക്സാമിന് ചോദിച്ച ഒരു ക്വസ്റ്റൻ ആണ്. വിച്ച് വൺ ഈസ് ക്വാണ്ടിറ്റേറ്റീവ് ആൻഡ് വിച്ച് വൺ ഈസ് ക്വാളിറ്റേറ്റീവ് ഡിസോർഡർ. Both are connected with the blood disorders. The last part of this chapter, that will be chromosomal disorders. Chromosomal disorders are also caused due to the absence or excess of abnormal arrangement of one or more chromosome. Absence or excess of abnormal arrangement. But the chromosome number will be affected. Chromosome number is called ploidy. Like normal chromosome number, it is called euploidy. If there will be either gain or loss in a set of chromosome, that is due to failure of segregation of chromatids. Failure of segregation of chromatids during cell cycle division, cell cycle division or cell division cycle results in the gain or loss of a chromosome. Gain or increase, or loss means decrease in a chromosome. That is called aneuploidy. That's called aneuploidy. അപ്പോൾ നമ്മൾ ഇതിനെ നമ്മൾ വിളിക്കുന്ന പേരാണ്, it's called failure of segregation of chromatids. It is called non-disjunction of chromosome. അതിനെ നമ്മൾ വിളിക്കുകയാണ് non-disjunction of chromosome. Non-disjunction of the chromosome. Failure of segregation of chromatids during cell division cycle results in the gain, increase or loss of a chromosome is called aneuploidy. അപ്പോൾ ചിലപ്പോൾ ഒരു സെറ്റിൽ കൂടുതൽ വരാം, ഒരു സെറ്റിൽ കുറയാം, അല്ലെ? either gain or loss. For example, Down syndrome നമുക്കൊരു gain ആണ്, result in the gain of an extra copy of chromosome in 21st chromosome ഒരെണ്ണം കൂടുമ്പോൾ അതിനെ വിളിക്കുന്ന പേരാണ്, that is called Down syndrome. Similarly, in the case of Turner syndrome, അപ്പോൾ നമ്മൾ ഡൗൺ സിൻഡ്രോമിൽ ഒരെണ്ണം കൂടുമ്പോൾ അല്ലെ 21st ക്രോമസോമിൽ ഡ്യൂപ്ലിക്കേറ്റിന് വരും, ട്രിപ്ലിക്കേറ്റ് വരും. അപ്പൊ നമുക്ക് ഓട്ടോസോമിന്റെ എണ്ണം കൂടും. ഓട്ടോസോം നോർമലി 44 വരുന്നതിന് വരെ എത്ര വരും? 45 ഓട്ടോസോംസ് വരും. 45 autosomes and plus the male or female. If it is female, XX. If it is male, XY. So the total chromosome is 45 A plus XX or XY. It will be 47 instead of 46. So extra copy of chromosome at 21. Adana Down syndrome. It is called trisomy. The gain is called trisomy. It is called 2n plus 1. 2n plus 1, about 45A. Similarly, Turner syndrome, another syndrome called Turner syndrome, result due to the loss of an X chromosome in human females. Loss of an X chromosome in human females. Turner syndrome due to loss of an X chromosome in human female. About human female, one X chromosome loss. So that is 44A plus X0. That is 45. അപ്പൊ അതിനെ നമ്മൾ എന്താ വിളിക്കുക? മോണോസോമി. ലോസ് വരുമ്പോൾ അതിനെ നമ്മൾ വിളിക്കുന്ന പേരാണ് മോണോസോമി. അല്ലെ, ഗെയിൻ വരുമ്പോൾ നമ്മൾ അതിനെ എന്താ വിളിക്കുക? ട്രൈസോമി. ലോസ് വരുമ്പോൾ നമ്മൾ എന്താ വിളിക്കുക? മോണോസോമി. അപ്പൊ 2n minus 1. 2n minus 1. 2n minus 1. അല്ലെ, അപ്പൊ ടർണർ സിൻഡ്രോം result due to the loss of an X chromosome in human females. അപ്പൊ അത് സെക്സ് ക്രോമസോമിൽ ഒരെണ്ണം കുറഞ്ഞു, XX പകരം X0 ആയി. X0 അല്ലെ. അതുപോലെ തന്നെ അപ്പൊ അതിനാണ് നമ്മൾ ടർണർ സിൻഡ്രോം. അപ്പൊ ഇതാണ് നമ്മൾ അന്യൂപ്ലോയിഡി എന്ന് പറയുന്നത്. ഐദർ ഗെയിൻ ഓർ ലോസ് ഓഫ് എ ക്രോമസോം ഡ്യൂ ടു ഫെയിലിയർ ഓഫ് സെഗ്രിഗേഷൻ ഓഫ് ക്രോമാറ്റിഡ്സ് ഓർ നോൺ ഡിസെൻഷൻ ഓഫ് ക്രോമസോം. അപ്പൊ എനി സെറ്റ് ഓഫ് ദി ക്രോമസോം ദർ വിൽ ബി ആൻ ഗെയിൻ ഓർ ലോസ്. അതിനൊരു എക്സാമ്പിൾ ആണ് ഡൗൺ സിൻഡ്രോം പറഞ്ഞ. വേറെ സിൻഡ്രോം നമ്മൾ ടർണർ സിൻഡ്രോം അല്ലെ. ഇന്നൊരു സിൻഡ്രോം നമ്മൾ കാണുന്നത് ക്ലിൻഫെൽറ്റർ സിൻഡ്രോം ആണ്. ക്ലിൻഫെൽറ്റർ സിൻഡ്രോമിൽ നമുക്കറിയാം, അത് നമ്മൾ അടുത്ത് പഠിക്കാൻ പോവാണ്. ക്ലിൻഫെൽറ്റർ സിൻഡ്രോമിൽ അവിടെ എന്താ സംഭവിക്കുന്നത്? There will be an additional X chromosome. In males there will be an additional X chromosome. അല്ലെ. അപ്പൊ നമുക്ക് ക്ലിൻഫെൽറ്റർ സിൻഡ്രോമിൽ നമ്മൾ എങ്ങനെയാണ്? 44A plus XXY. That will be 47. അപ്പൊ there will be an gain. It is also trisomy. This is also trisomy. അല്ലെ, trisomy. 2n plus 1. പക്ഷെ ഇവിടെ ട്രൈസോമി നടന്നത് എവിടെയാ? സെക്സ് ക്രോമസോമിലാണ് നടന്നത്. ഇവിടെ ട്രൈസോമി നടന്നത് എവിടെയാണ്? That is in the case of ഡൗൺ സിൻഡ്രോമിലെ ഓട്ടോസോമിലാണ്. That will be in autosome. അപ്പൊ നമ്മൾ അതിനെ ഓട്ടോസോമൽ ട്രൈസോമി എന്ന് വിളിക്കും. ഇവിടെ അതിനെ സെക്സ് ക്രോമസോമൽ ട്രൈസോമി എന്ന് വിളിക്കും. അത് നമ്മൾ അടുത്ത് പഠിക്കാൻ പോകുന്നുണ്ട്. അപ്പൊ അറിയാം, failure of cytokinesis after telophase stage of the cell division results in an increase in the whole set of chromosome in an organism, and this phenomenon is known as polyploidy. Failure of cytokinesis. Karyokinesis, division of nucleus, is followed by cytokinesis, division of cytoplasm. Failure of cytokinesis after telophase. Telophase means the last stage of karyokinesis. അപ്പൊ telophase കഴിഞ്ഞ് കഴിയുമ്പോൾ cytokinesis നടക്കാതെ വന്നു കഴിഞ്ഞാൽ results in the increase in the whole set of chromosome. Chromosomes that get multiplied or doubled. Doubling of the chromosome or multiply the chromosome. This phenomenon is called polyploidy, and it is often seen in plants. Polyploidy is often seen in plants. About the regular increase in the every set of the chromosome. Polyploidy either gain or loss in any set of the chromosome. In any set of the 22 pairs, 22 that is 22 pair ഏതെങ്കിലും ഒരു pair either increase or decrease വരികയാണെങ്കിൽ അതിനെ അന്യൂപ്ലോയിഡി എന്ന് വിളിക്കും. ഗെയിൻ ഓർ ലോസ്. ഇവിടെ അങ്ങനെയല്ല. ഇവിടെ റെഗുലർ ഇൻക്രീസ് ഇൻ ദ ഹോൾ സെറ്റ് ഓഫ് ക്രോമസോം. എല്ലാ സെറ്റിലും ഹോൾ സെറ്റ് ഓഫ് ക്രോമസോം കൂടുമ്പോൾ നമ്മൾ വിളിക്കുന്ന പേരാണ് പോളിപ്ലോയിഡി. അല്ലെ, ദിസ് കണ്ടീഷൻ ഇസ് ഓഫൺ സീൻ ഇൻ പ്ലാന്റ്സിലാണ് കാണുന്നത്. ഓക്കെ, ഇനി ഇത് അടുത്ത് നമ്മൾ ടോട്ടൽ നമ്പർ ഓഫ് ക്രോമസോം ഇൻ എ നോർമൽ ഹ്യൂമൻ സെൽ ഇസ് 46. That is 23 pairs. നമുക്കറിയാം, out of these 22 pairs are autosomes. Only one pair of the chromosome will be sex chromosome. Sometimes, though rarely, either an additional copy of a chromosome may be included in an individual. Either an additional copy of chromosome may be included in an individual. ഒരു കോപ്പി ആഡ് ചെയ്ത് കഴിഞ്ഞാൽ any set of the chromosome, or loss of an or individual may lack one pair of the chromosome. Either gain, addition, or may lack one of any pair of the chromosome. These situations are called add addition trisomy, or it may lack one of any one pair of chromosomes. That's called monosomy of a chromosome. Such a situation leads to various serious consequences in the individual. In the syndromes are chromosomal syndromes, chromosomal disorders, Down syndrome, Turner syndrome, and Klinefelter syndrome. Common examples of chromosomal disorder. Down syndrome patient represent a figure showing an individual infected with the Down syndrome and corresponding the chromosomes of the individual. Chromosome diagram, ideogram. Diagrammatic representation of the chromosome, karyotypes is called ideogram. But Down syndrome, this cause was genetic disorder, is the presence of an additional copy of the chromosome number in 21, and it is called trisomy of 21. Disorder are described by the scientist Langdon Down. Langdon Down. Down syndrome. Down syndrome, 1866. The affected individual is short-statured with a small round head. Small round head, round head and furrowed tongue, and partially open mouth, and palm is broad with a characteristic palm crease. There will be only single crease, single line, palm line. That will be transverse line. Palm crease, specific palm crease. No, there is only a single transverse palm crease. That is the specific palm crease. Only a single crease. Transverse line. There is a transverse. Specific palm crease. Specific with the palm crease. Palm is broad with the characters palm crease. Physical, psychomotor, and mental development is retarded. Down syndrome. Down syndrome. Down syndrome patient 21st. 21st chromosome. Duplicate duplicate 1, 2, 3. Additional copy is added in the 21st chromosome. This is the 21st chromosome. Ideogram. Diagrammatic representation of chromosome is called ideogram. Okay. Down syndrome. Physical, psychomotor, and mental development is retarded. Then second one is Klinefelter syndrome. That is the case of this one. Klinefelter syndrome. This genetic disorder is also caused by the presence of an additional copy of X chromosome. Additional copy of an X chromosome resulting into a karyotype of 47. Karyotype of 47. XXY. Such an individual has an overall masculine development. Masculine development. Characters masculine characters development masculine. But however, the feminine development, feminized characters, that is development of breast development in males. Gynecomastia. That is called gynecomastia. Feminine development. Underdeveloped testis. Testis underdeveloped. Sterile. A person tall stature with the feminized characters. Body hair body hairs that is the case of more feminine characters. Resulting in a karyotype of 47. XXY condition. Additional X chromosome. About trisomy and additional X copy of X chromosome 47. XXY plus 44A plus XXY, that is 47 instead of 46. Overall masculine development and feminine development development of breast development males. Gynecomastia, and such individuals are sterile. Okay. Then next one is called Klinefelter syndrome is a trisomy. Allosomal trisomy sex chromosomal trisomy. Down syndrome autosomal 21 autosomal trisomy. Okay. In Turner syndrome, such a disorder is caused by the absence of one of the X chromosome in female. 44A plus X0, that is 45 with the X0. Such females are sterile. Females are sterile. And their ovaries are rudimentary. Their uterus is undeveloped. Breast undeveloped breast. Underdeveloped breast, ovary, no menstruation. That is the case of ovaries are rudimentary. Rudimentary not developed. Uterus short stature, webbed neck. Webbed neck. Features lack of other secondary sexual characters. lack of other secondary sexual characters. അപ്പോൾ symptoms of Turner syndrome. Turner syndromeന്റെ symptoms. അപ്പോൾ അവിടെ മെയിൻ പോസിറ്റീവ് എന്താണ്? There will be an monosomy. Monosomy എന്ന് പറഞ്ഞാൽ എന്താ? Loss of an X chromosome. നോർമൽ ഫീമെയിലിൽ XX വരുന്നതിന് പകരം ഇവരിൽ എന്ത് വരുള്ളൂ? Only one X chromosome. Lacking one X chromosome. അപ്പോൾ that is 44A plus XX ന് പകരം 44A plus X0. അപ്പോൾ 46 ക്രോമസോമിന് വരെ എത്ര ക്രോമസോമാ? 45 chromosome. അപ്പോൾ it is a monosomy and it is an aneuploidy. It is an aneuploidy and it is monosomy. Okay. അപ്പോൾ Klinefelter syndrome trisomy. അത് രണ്ടും ഏത് ക്രോമസോമിലാ? സെക്സ് ക്രോമസോമിൽ. ഡൗൺ സിൻഡ്രോം നടക്കുന്നത് ഓട്ടോസോമിൽ. ഓട്ടോസോം 21st ട്രൈസോമി. അപ്പൊ 21ാമത്തെ ക്രോമസോമിൽ ഒരെണ്ണം ട്രിപ്ലിക്കേറ്റ് വരും, കൂടുതൽ വരും. 21st അല്ലെ? 21. ബാക്കി എല്ലാത്തിലും പേഡ് രണ്ടെണ്ണമാണ് വരാം അല്ലെ? ഇവിടെ മാത്രം 21st 3 ക്രോമസോം. അഡീഷണൽ കോപ്പി വിൽ ബി ഇൻ ദ 21st and it is in the autosome. അപ്പൊ അതിനെ നമ്മൾ ഓട്ടോസോമൽ ട്രൈസോമി. ഓക്കെ. അപ്പൊ ദീസ് ആർ ദ ഇമ്പോർട്ടന്റ് ക്രോമസോമൽ ഡിസോർഡേഴ്സ് and this chapter is over. But this is only a brief account of the chapter. That is in the case of you have to complete this chapter in 10 hours or more than 10 hours, more than 12 hours. 12 hours. That will be taken for only 3 hours. But that is only a briefing, only brief accounting. Okay. Thank you. Thank you, everybody. Allah. Thank you.